Can we start? Okay, very good. So very good morning to all of you and welcome to this marathon gold class of botany first. Okay, so I have decided to be starting with the physiology topics. Instead of starting with the living world, I'll be starting with the physiology topics. Is it okay with everyone? Fine. So we are starting with photosynthesis in higher plants. Fine. And a very, very basic definition of photosynthesis is photosynthesis is the process in which green parts of plants combine inorganic raw materials like CO2 and water in presence of sunlight to synthesize organic food like glucose. Fine, very basic simple definition. So we can say 6 CO2 plus 12 H2O, chlorophyll, sunlight, C6H12O6 plus 6 O2 plus 6 H2. Basic definition, fine. So this is the basic definition of photosynthesis. So inorganic, that is, it is a type of autotrophic mode of nutrition, fine. Okay. Now let us see, if we see the photosynthesis as a process. First, it is anabolic process. Why anabolic process? Because there is synthesis of food. Synthesis of something through a biochemical reaction is called as anabolism. So it is an anabolic process. Second, endergonic process. Why? Because energy is being used in this process. The energy of sunlight is being used. So it is an anabolic process. It is an endergonic process. Third, it is an oxidation reduction process. Why? Because water is oxidized while CO2 is reduced. It is directly or indirectly source of food for all organisms living on this earth. Okay, herbivores directly and carnivores indirectly. So it is source of food. Fifth, source of O2 for aerobic organisms. Because when water is oxidized, the oxygen is released from this water into the atmosphere, and this forms the atmospheric oxygen, which is the source of oxygen for aerobic organisms. So these are the basics of the photosynthesis and as a process. Let us see now, what do we already know? Let us see. What do we know? First of all, a variegated leaf experiment. So see, a variegated leaf is a leaf where chlorophyll is not evenly distributed throughout the leaf. In certain patches chlorophyll is present, at other places is not present. And if you cover this leaf by a black paper and then you expose to photosynthesis, you see two things. First, production is seen only at those places where chlorophyll was present in the leaf. So first of all, this proves that chlorophyll is essential for photosynthesis. And second, part which was covered by black paper, at that part there is no starch synthesis. It means light is also essential for photosynthesis. So this experiment information. First, chlorophyll is essential for photosynthesis and light is also essential for photosynthesis. Fine. So variegated leaf that was partially covered with black paper and exposed to light. On testing the leaves of the presence of a starch, it was clear that photosynthesis occurs only in the green parts of the leaf and only in the presence of light. So with this experiment, two things we prove. First, the importance of green part, that is chlorophyll, and second, the importance of light. Is it clear? Second, a part of leaf is enclosed in a test tube containing cuvette that soaks, absorbs CO2. So this is called as Mohs half leaf experiment. So half leaf is put in part of the leaf was in synthesis. So this proves that CO2 is essential for photosynthesis. So Mohs half leaf experiment proves that CO2 is essential for photosynthesis. So three things we have said. Chlorophyll is essential, light is essential, and CO2 is essential. Fine? Okay. Next. So these are the some we know. Now early experiments. Now see early experiments. Joseph Priestley. Joseph Priestley, what he did, he first of all placed a mouse inside a bell and a burning candle. So burning candle was using the oxygen inside and the mouse was also using the oxygen inside for respiration. And after some time, when all the oxygen was over, voice is breaking. Okay, I'll see. Fine, okay. They're saying voice is breaking. There's some complaint that voice is breaking. Is it breaking for everyone? Voice is breaking, they are saying. Okay. We are trying to fix it, don't worry. Hello? Hello, can you hear me? What happened? Okay, can you hear me now? Voice is clear now? Okay, fine. Fine, voice is clear now. Okay. Okay. Okay, thank you. Thank you. Thank you. Fine. So, okay. See, so Joseph Priestley, he also remembered, he also discovered oxygen, Joseph Priestley. So Joseph Priestley, what he did, he placed a burning candle and a breathing mouse inside a bell jar, and after some time, when all oxygen was consumed, the candle was put off and the mice died. Okay? In a repeat experiment, what he did, along with the burning candle and the breathing mouse, he plant inside. Okay, and this time neither did the mice die nor was the candle put off. So Joseph Priestley said that plants have the ability to purify the air. Plants can purify the foul air into fresh air. So this was, so importance of air was given by Joseph Priestley. Second is Jane Ingenhouse. Jane Ingenhouse repeated the same experiment, but he put a plant in water covered by a funnel. So first of all, three things he said. He said when plants perform photosynthesis, bubbles that come out, they are of oxygen. So he told that oxygen is released during photosynthesis by plant, first thing. Second, plants can purify air, this is correct, but only in presence of sunlight. If the same experiment is performed in dark, there is no air purification, there is no release of oxygen. So second, he told the importance of the light. Third, that only green parts of the plant can purify the air. Non-green parts of the plant cannot purify the air. So he also told the importance of chlorophyll. So three things he said. First of all, the oxygen released during the gas released during photosynthesis is oxygen. Second, plants can purify air only in presence of light. And third, only green parts of the plant can purify the air. All 3 points clear? Next. Julius von Sachs. Julius von Sachs, he gave certain evidences, first provided evidence for production of glucose when plants grow. So he was the one who told what is the product of photosynthesis, and he said the product of photosynthesis is glucose. But plants stored this glucose in the form of starch, okay? And you know starch is a homopolymer of glucose. So glucose is usually stored in the form of starch. So first point, glucose. Second, glucose is stored in the form of starch. Third point, he said that the green substance in plants, now we know chlorophyll is located in special bodies, and these special bodies now we call as chloroplast. So he also told that the chlorophyll is present in the chloroplast. He found that green parts in plant is where glucose is made, and that glucose is usually stored as starch. So these 3 points were done by him. So 3 things by him. First of all, that plants produce the product of photosynthesis, glucose. Second, it is stored in the form of a polymer starch. Third point, that the green pigment that is responsible, that is present inside the structure which we now call as the chloroplast. Fine. Next. This is very important. T.W. Engelmann. You know, the mixture of all, if we can, now plant form 700 nm. By the prism T.W. Engelmann, he separated the different wavelengths of light. Clado filamentous green alga which is performing photosynthesis. When oxygen is released, let us see why this problem is early. Okay, let me see. Again, problem. You see. Again, they are saying voice problem. You remain here, no? You remain here for some time. Is it okay now? Is the voice okay now? Okay. Let us hope it remains okay. Now onwards it remains okay. Fine? Okay. Let us continue. Fine. So he used a filamentous green alga cladophora and he used aerobic bacteria, and he, as I said, he separated the different wavelengths of light by using a prism, and he found that maximum photosynthesis takes place in blue and red light. This is called as the action spectrum of photosynthesis. So first action spectrum of photosynthesis was given by W. Engelmann by using cladophora, a filamentous green alga, and aerobic bacteria, and he proved that maximum photosynthesis takes place in blue and red light. This point is clear, and this is called as the action spectrum of photosynthesis. Later on, we will discuss this spectrum also in detail. Fine. Next. So by the middle of the 19th century, the summary equation of photosynthesis was known, that CO2 plus water is equal to glucose plus O2. Now the question was, the oxygen that is being released in photosynthesis, whether this oxygen is coming from CO2 or it is coming from water. This was not clear. The oxygen released in photosynthesis, what is the source of this oxygen, or CO2 or water? An indirect hint of this was given by Carolus von Niel. Okay, so next is Carolus von Niel. So Carolus von Niel, he said that photosynthesis is an oxidation reduction process, fine, where a hydrogen donor is there that reduces the CO2 to produce glucose. Now, depending upon the chemical nature of the hydrogen donor, it is seen that oxygen is released or not released. So he performed photosynthesis in purple and green bacteria. So in purple and green bacteria, the source of hydrogen is the source of hydrogen is H2S, and he saw that now oxygen is not released. Okay, so he said that in purple and green bacteria which do not use water as hydrogen source, they use H2S hydrogen source. So the oxygen is not released, instead sulfur is released. On the basis of this, he said, since plants use water as hydrogen source, that is why in plants the oxygen is released. It means the oxygen released in photosynthesis comes from water and not from CO2. Okay, so by this his experiment, by indirectly he proved that the oxygen released in photosynthesis comes from water and not from oxygen. Okay, a more direct proof was given by Ruben-Kamen and Kamen. They used O18, a isotope of oxygen. So they saw that if this isotope of oxygen was used in CO2, the released oxygen was O16, means there was no radioactivity in the released oxygen. But when radioactive oxygen was used in water, the released oxygen was radioactive O18. So this gave the direct proof that the oxygen released in photosynthesis comes from water and not from CO2. Is this point clear to everyone? Hello? Okay, fine. So now, after knowing that the oxygen released in water, oxygen released in photosynthesis comes from water, and for production of 1 glucose, 6 water molecules, 6 oxygen is released. So how many water molecules will be required? See here. Now, if this oxygen is coming from water, so to release 6 oxygen molecules, minimum water molecules required will be 12, because NCERT has asked this question to you. Okay, why if 6 water is finally come out, then why 12 water molecules are used? 12 water molecules are used because for release of 6 oxygen, the minimum water molecules will be required is 12. That is why it is C6CO2 plus 12 H2O is equal to C6H12O6 plus 6O2 plus 6H2O. Is it clear? So this is all regarding the early experiments. So you can see it like this. Okay. So all the oxygen of water is released in the form of this. And if you are asked where does the oxygen of CO2 go? So oxygen of CO2 goes into the glucose. So both questions can be asked. If it is asked where does the oxygen of water go? In the atmosphere, released oxygen. Where does the oxygen of CO2 go? It goes into the glucose. Fine. So this is all regarding the early experiments. So that is why this reaction is like this. 12 water molecules are used. Now, where does photosynthesis takes place? So remember, in plants, there is a special organelle which is called as chloroplast. So in chloroplast, photosynthesis takes place in plant. Now, this chloroplast does not have a fixed position. Chloroplast is generally found in the mesophyll cells of the leaves. Okay, chloroplast is generally found in the mesophyll cells of the leaves. But there are exceptional cases also. For example, plants like Opoecia, chloroplast develops in the stem. Okay. In certain plants like Ctenophora, chloroplast develops in the root also. So there are some exceptions, but generally chloroplast is present in the mesophyll cells of the leaves, and the position of the chloroplast in mesophyll cell of leaf is not fixed. It depends upon the incident light, intensity of the incident light. If the intensity of the incident light is low, if the intensity of the incident light is low, suppose this is the incident light, then the chloroplast, flat surface of the chloroplast is perpendicular to the incident light at low light intensity, and this condition is called as epistrophe. But if the intensity of the incident light is very high, if the intensity of the incident light is very high, so to protect it from the photooxidation, remember in very high light intensity, there is oxidation of the chlorophyll, which is called as destruction of the chlorophyll, called as photooxidation. So protect it from the photooxidation, the flat surface of chloroplast, it comes parallel to the longitudinal wall. Okay, this is called as apostrophe. Sorry, parastrophe. So depending upon the incident light, the position of the chloroplast in the cell in the mesophyll cell can shift from one place to another place. Fine. This question has also been asked in your NCERT, and this is the answer to this question. Fine. Now, if we see the structure of chloroplast, chloroplast is a double membrane bound structure. Okay, and interior of the chloroplast has two parts. Interior of the chloroplast has two parts: a membranous part called as thylakoids. Okay, and a fluid part called as stroma. Now, these thylakoids also are two types. If many thylakoids are stacked one over the other, this is called as grana. Okay, and if thylakoids are not stacked, as you can see this portion, not stacking over other, then this is called as stroma lamella. Okay, so thylakoids are of two types: stacked thylakoid, this is called as grana, and unstacked thylakoid, this is called as the stroma lamella. Whether it is grana or whether it is stroma lamella, both are the site of light reaction of photosynthesis. And you know, for light reaction, the chlorophyll pigment and other photosynthetic pigments will absorb light. That is why in this thylakoid, if you see thylakoid, it is something like this, a hollow structure. And in the membrane of thylakoid, there are complexes called as pigment system. And in this pigment system, protein pigment complexes are present, which absorb the sunlight to initiate the light reaction of photosynthesis. And this hollow space is called as the lumen of the thylakoid. Fine. So if thylakoids are stacked, grana, unstacked, stroma lamella, and both are site of light reaction of photosynthesis. Fine. And the stroma part, this stroma part, this is the site of dark reaction of photosynthesis. Now, what are the products of light reaction and dark reaction? During light reaction, due to light absorption, there is ultimately synthesis of two compounds, ATP and NADPH. Both ATP and NADH are the direct product of light reaction, and they are used in the dark reaction to reduce CO2 to produce glucose. Okay. So, is it appropriate to call it dark reaction? Does it take place in dark? No, it cannot take place in dark because it is dependent on the products of light reaction. So just to separate it from the light reaction, we call it as dark reaction, but it does not mean that it can independently take place in the dark. Why? Because it is dependent on the products of the light reaction. Fine. So this is all regarding the chloroplast. Now, if we come to photosynthetic pigments, photosynthetic pigments in higher plants are chlorophyll A, chlorophyll B, and carotenoids. Carotenoids are of two types: carotenes and xanthophylls. Chlorophyll A is bluish green in color and it is a universal pigment. Except the purple and green bacteria, all photosynthesizers have chlorophyll A. So that is why chlorophyll A is the universal pigment, and chlorophyll A is the chief photosynthetic pigment. All other pigments are called as accessory pigments. Second, chlorophyll B, it is yellow-green in color. It is an accessory pigment. It is not found in all. It is found in plants, higher plants. It is found in green algae, and it is also found in euglenoids. That is why we say euglenoids have photosynthetic pigments similar to the higher plants. Carotenoids, carotenes are yellow to yellow-orange, and xanthophylls are yellow. And these carotenes and xanthophylls are also accessory pigments. So remember, except chlorophyll A, all other pigments are accessory pigments. And now these pigments absorb different wavelength of light, but within the range that is 400 to 700 nm, which is called as the visible light. If I make a graph to show which pigment absorbs which wavelength of light maximum, this graph is called as the absorption spectrum. So let us see the absorption spectrum. See, This graph is absorption spectrum. This is the wavelength of light. Okay, this is the wavelength of light, 400 nm, 500 nm, 600 nm and 700 nm. Okay, and this is percentage absorption. So first of all, we start with chlorophyll A. The blue line is showing chlorophyll A. So you see chlorophyll A is maximum absorbing blue light, and again it is absorbing red light. So chlorophyll A absorbs maximum blue and red light. So this is the absorption spectrum of chlorophyll A. Now we come to chlorophyll B, which is the green line. Again, chlorophyll B is also absorbing maximum blue light and again red light. So both chlorophyll A and chlorophyll B absorb maximum blue and red light. Now chlorophyll B absorbs more blue light as compared to chlorophyll A, as you can see here, but chlorophyll A absorbs more red light as compared to chlorophyll B. Now carotenoids, if we see this yellow one is carotenoids, they absorb light between 400 to 500 nm. Okay, so they absorb such wavelength of light which is not absorbed by chlorophyll A or chlorophyll B. So first function of carotenoids is an accessory pigment to absorb that wavelength of light which is not used by chlorophyll A or chlorophyll B. So they increase the range of light that can be used in photosynthesis. Okay, so this is the first advantage of carotenoids. But carotenoids also have one more advantage. During the light reaction of photosynthesis, in presence of light, water is split into 2 protons plus 2 electrons plus nascent oxygen. This nascent oxygen is very, very reactive and it causes oxidation of the chlorophyll, which is called as photooxidation of the chlorophyll, causing damage to the chlorophyll. Here carotenoids have important role. Carotenoids convert this nascent oxygen into molecular oxygen and protect the chlorophyll from photooxidation. So there are 2 roles of carotenoids. First, to function as accessory pigment to increase the range of wavelength of light that can be used in photosynthesis. And second, they protect the chlorophyll from photooxidation by converting this nascent oxygen into molecular oxygen. So this is all regarding the various photosynthetic pigments. Is it clear to everyone? Okay. Now if you see a leaf, it appears green, maybe different shades of green, dark green, some yellowish green, because of the different because of the different proportions of different pigments. Now if you want to separate different pigments of chlorophyll in the leaf, okay, crush the leaf, take a leaf, but generally we take a spinach leaf, palakura, we crush it and then we subject it to a process called as paper chromatography. Paper chromatography. And on the basis of this paper chromatography, the basis is the molecular weight. More the molecular weight, lesser distance it will travel. So on this basis, by paper chromatography, we can separate different pigments present in a leaf by this phenomena. Fine? Okay. So different colors you can see. And how can you identify? By the colors? Because the color of different pigments is slightly different from each other. That is why we can operate it. Fine? Okay. So at lowest will be chlorophyll B, because chlorophyll has the maximum molecular weight. Then chlorophyll A. Okay. Then carotenes and then xanthophylls and then carotenes. And then carotenes. So this is on the basis of their molecular weight, the different photosynthetic pigments can be separated by a process called as paper chromatography. Fine. Next we come to another thing. Remember Engelmann's work. We said that T.W. Engelmann gave the action spectrum of photosynthesis. So what is an action spectrum? A graph that shows efficiency of various wavelengths of light in influencing the process of photosynthesis. Means in which wavelength of light maximum photosynthesis takes place, in which wavelength of light minimum photosynthesis takes place, and on the basis of this, if we make a graph, this graph is called as action spectrum. And you know this was given by T.W. Engelmann, and he said that maximum photosynthesis takes place in blue and red light. So let us see this graph. See. So this is the graph again. Here, how will you measure that how much photosynthesis has taken place? So very simple measure is amount of O2 released. More the rate of photosynthesis, more the O2 will be released, and this is how Engelmann discovered the rate of photosynthesis. So how much photosynthesis is, how much oxygen is released in which wavelength of light, this gives an indirect measure of the rate of glucose production. So here the amount of O2 released, and here the wavelength of light, again 400, 500, 600 and 700. So when this was done, it was seen that maximum O2 released in the blue light. Means maximum photosynthesis takes place in blue light, and again maximum O2 released in red light. It means maximum photosynthesis takes place in the red light. So this is the action spectrum of photosynthesis, which shows if plant is exposed to monochromatic light, single colored light, then maximum photosynthesis takes place in blue and red light. And remember, absorption spectrum said that chlorophyll A absorbs maximum blue and red light. So now let us compare the absorption spectrum of chlorophyll A and action spectrum of photosynthesis. So this is the third graph. See here. The black line shows the action spectrum of photosynthesis, and blue line shows the absorption spectrum of chlorophyll A. You see that the two graphs are nearly superimposing each other, nearly overlapping. I'm not saying completely overlapping, but nearly overlapping each other. So from this, we come to the conclusion that chlorophyll A absorbs maximum blue and red light, and maximum photosynthesis is taking place in blue and red light. It means the chief photosynthetic pigment is chlorophyll A. Let us see this conclusion here. Can you see that the wavelength at which there is maximum absorption by chlorophyll A, that is in the blue and the red region, also shows higher rate of photosynthesis. Hence, we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. Fine. But you see, there is not complete overlapping. Here, there is not complete overlapping. So this not complete overlapping because some photosynthesis is also carried out by the accessory pigments. Because some photosynthesis is also carried out by the accessory pigment, it means in those wavelengths which chlorophyll A is not absorbing, some photosynthesis is taking place due to the action of the carotenoids. That is why the two graphs are not completely overlapping each other. This point is clear, children? Anyone wants to ask any question? Come on. Come on, fast. Okay, fine. Now see. Photosynthesis as a process. So photosynthesis has two phases. First, light reaction or photochemical phase. Okay, light reaction or photochemical phase. In light reaction or photochemical phase, let us see what happens. Light absorbed by pigments, excitation of pigments, release of electron, movement of electron on electron transport system. Due to this synthesis of NADPH plus H plus and ATP. So these are the products of light reaction. That is why light absorption is must for synthesis of NADPH and ATP. Both are together called as assimilatory powers. But if the process has to continue, because chlorophyll has released its electron, it has become oxidized. But if the process has to continue, there should be someone to donate electron to chlorophyll so that the process can continuously take place. So along with this, there is again splitting of water to release electron, accepted by chlorophyll. Fine. So now the process can continue. Chlorophyll will get excited, release electron, electron will move to electron transport chain. As a result, there will be synthesis of NADPH and ATP. And the electron loss of the chlorophyll will be fulfilled by the oxidation of water. Okay. When water will be oxidized, I said there will be release of O2. So if we see the products of light reaction, there are three products of light reaction. First, NADPH, second ATP, and third O2. O2 will be released into the atmosphere, while NADPH and ATP are immediately used in the next phase, which is called as the dark reaction. And I've already told you the site for this light reaction is the thylakoid, that is both grana and stroma lamni. So first part is the photochemical phase or the light reaction. Second is dark reaction or biochemical phase. The site is stroma, which has enzymes. By using ATP and NADPH plus H plus produced in light reaction, CO2 is reduced to synthesize glucose. As I said, it is not directly dependent on light. That is why we call it as dark reaction. But it is not very proper to call it dark reaction. Why? Because it cannot independently operate in dark, because it is dependent on the products of light reaction. So these are the two phases. So first of all, let us start with what is light reaction. What is light reaction? So remember, we said that first phase in photosynthesis, if you are asked what is the first phase in photosynthesis, absorption of light by the photosynthetic pigments and their excitation to release electron. Now, single chlorophyll molecule or single pigment molecule cannot perform this process. So in the thylakoid membrane, there are pigment protein complexes which are called as pigment systems or photosystems. One pigment system or photosystem consists of around 250 to 400 pigment molecules. And now this pigment system has two parts. This pigment system has two parts. One part is called as reaction center. It is a single chlorophyll A. Single chlorophyll A molecule, which is called as a reaction center, and all other pigment systems are called as antenna molecules, or they are also called as light harvesting complex. Light harvesting complex. Okay. So a pigment system consists of two parts, a single chlorophyll A molecule called the reaction center, and all other which may have chlorophyll A, chlorophyll B, carotenoids, all, they are collectively called as antenna molecules, or they are called as a light harvesting complex. Now what happens when the light falls, photons means packets of light, they fall on the antenna molecules. The antenna molecules get, due to this light absorption, get excited, and when they get excited, they resonate. And when they resonate, they collide with each other. And when they collide with each other, they transfer their energy to the next one. So this pigment will transfer energy to this to this, and ultimately all this energy is given to the reaction center. So remember, first thing is excitation of the antenna molecules. Due to this, they release due to energy, they resonate, and due to resonance, they collide with each other and they transfer their energy. And ultimately all this energy is transferred to the reaction center. And now reaction center gets so much energy that it loses an electron to a primary acceptor. Fine, it loses electron to a primary acceptor. And from primary acceptor, the electron moves on a electron transport chain, which ultimately results in the synthesis of ATP and NADPH. This point is clear. This is very, very important point. If you understand this, then only you will understand the light reaction. So again, I am repeating. The members in the antenna molecule, they absorb sunlight, they get energized, and due to this energy, they resonate. When they resonate, they collide with each other, and due to this, they transfer their energy to ultimately to the reaction center. This is called as resonance transfer. Reaction center gets so much energy that it loses an electron to a primary acceptor. Now this electron moves on a chain of electron carrier to ultimately synthesize the ATP and NADPH. Fine. Now, and this is, I said, this complete is called as a pigment system. Now it was discovered that later on there are two types of pigment systems. Pigment system 1 and pigment system 2. This was discovered earlier, and this was discovered later. So remember, the name PS1 and PS2 is not on the basis of their order of their functioning. The name PS1 and PS2 is on the basis of their order of discovery. Okay. Here the reaction center is a chlorophyll A that absorbs light of 700 nanometer. That is why it is called as P700. And here the reaction center is a chlorophyll A that absorbs light of 680 nm. That is why it is called as P680. Okay. So P stands for pigment, pigment 700. Okay. Now, chlorophyll pigments, if you see the thylakoid membrane, both pigment systems are embedded in the thylakoid membrane, but PS1 is slightly towards the stroma side, while PS2 is slightly towards the lumen side. So their positioning is slightly different. PS1 is towards the stroma side of the thylakoid membrane, while PS2 is towards the lumen side of the thylakoid membrane. Fine. Okay. So this is PS1 and PS2. PS2 is mainly absorbed in splitting of water. While PS1 is mainly absorbed in reduction of NADP to NADPH. So PS2 is mainly involved in oxidation of water, while PS1 is mainly absorbed in reduction of NADP to NADPH. Fine. So this is all regarding. Now let us try to see the electron transport system. Electron transport system, or it is called a Z scheme. It involves both PS1 and PS2. So see here. So here I have written P680, it means it is the pigment system 2. The LHC will absorb light and lose electron to a primary acceptor. The primary acceptor is called as pheophytin. Remember, the primary acceptor is called as pheophytin. This is PS second. This is called as the uphill journey of electron. This is called as uphill journey of electron. From here, electron moves downhill journey. Okay, from here it moves downhill journey. So remember, important point, the primary acceptor of PS2 is pheophytin, which is a modified form of chlorophyll. Fine. Now electron is taken by a mobile carrier plastoquinone, which is embedded in the thylakoid membrane. From here, electrons are taken by a complex which is called as cytochrome B6F complex. Okay. From here, the electrons are taken by another mobile carrier which is called as plastocyanin, and this plastocyanin moves on the inner side of the thylakoid membrane, towards the inner side of the thylakoid membrane. And this electron is ultimately accepted by the reaction center of the PS1, which is P700. But the question is, why this P700 should accept this electron? It accepts why? Because as PS2 has lost electron due to light absorption, PS1 also loses electron due to light absorption to a primary acceptor, okay, which is an iron sulfur complex, FeS complex. So since it is electron deficient, so it pulls the electron from the plastocyanin to get to the ground state. This is, so you see, this is uphill journey of electron, this is downhill journey. Again, this is uphill journey, and again downhill journey starts. From FeS, the electrons are taken by ferredoxin, and from ferredoxin, ultimately the electrons are taken by NADP plus, which gets reduced to NADPH. So this is again downhill journey of electron, and this process is catalyzed by an enzyme, ferredoxin NADP reductase. The enzyme is called FNR, ferredoxin NADP reductase. So you can see the characteristic shape is zigzag, that is why this is called a Z scheme. And so one product of light reaction has been produced, that is the NADPH. And also due to this downhill journey of electrons, energy becomes available. And this energy which becomes available, it is used to combine ADP and inorganic phosphate to form ATP. So both products of light reaction, ATP and NADPH, are produced. But one more thing, children, I told you that PS1 fulfilled its electron loss by pulling electron from PS2. How will PS2 fulfill its loss? So PS2 fulfilled its loss by splitting water. And this electron is taken by PS2. So this process keeps on continuing. So we can say that in the Z scheme, the ultimate electron acceptor is NADP plus, while ultimate electron donor is the water. And this process continues. Fine. So this Z scheme, it is called Z scheme. It involves both PS second and PS1. You can write here PS1. PS1. Fine. Is this clear to everyone? Anyone wants to ask any question on this? Fine. Okay. So first we have done this production of NADPH. Second is production of ATP, which is called as photophosphorylation. Photophosphorylation. Production of ATP by illuminated. Illuminated means on which light is falling, that is called illuminated. By illuminated chloroplast. This is of two types. First is called as non-cyclic photophosphorylation. Non-cyclic photophosphorylation, why we are calling it, let us try to see. We are calling it non-cyclic photophosphorylation, because during this ATP production, the electron that was released by PS2 never returns back to PS2. So that electron is ultimately taken by NADP+. So electron is not cycled back to the original donor. That is why we are calling it as non-cyclic photophosphorylation. Fine. In non-cyclic photophosphorylation, certain other features, along with ATP, there is also production of NADPH. And another thing, in non-cyclic photophosphorylation, both PS2 and PS1 are involved. Fifth point, in non-cyclic photophosphorylation, along with ATP and NADPH, there is also production of O2, or there is also splitting of water. Fine. So these are the features of non-cyclic photophosphorylation. Let us see once more. Why it is called non-cyclic? Because electron is not cycled back to its original donor. Second, along with ATP and NADPH is also produced. Third, there is also splitting of water. And fourth, it involves both PS1 and PS2. Another thing is cyclic photophosphorylation. Cyclic photophosphorylation. It involves only PS1. So see here. See, first of all, it involves only PS1. It does not involve PS2. Second, there is synthesis of only ATP. No NADPH is synthesized. So first, it involves only PS1. Second, only ATP is synthesized, no NADPH is synthesized. Third, there is no splitting of water. Okay. And fourth, why we are calling it cyclic? Because you see, the electron released from PS1 ultimately cycles back to PS1. That is why there is no need of an external electron donor. That is why it is called as a cyclic photophosphorylation. Now, where does it take place? This probably takes place in stroma lamellae. See, for non-cyclic photophosphorylation to take place, two things are required. There should be presence of PS second, and there should be presence of FNR enzyme. But in stroma lamellae, no PS second absent and FNR enzyme absent. So since stroma lamellae does not contain PS second and FNR enzyme, that is why the electrons ultimately return back to the PS1. So the site for cyclic photophosphorylation is the stroma lamellae, which lacks PS second and the FNR enzyme. Another thing, you know, the reaction center in PS1 is 700, means it can absorb light up to 700. While PS1, the reaction center is P680, it's PS2. It means PS2 can absorb maximum light of 680 nm. So if you give light of only more than 680 nm, so PS2 will stop working. Maximum PS2 can work at 680 nm. So if you give light of only more than 680 nm, PS2 will stop working, then only phosphorylation possible will be the cyclic photophosphorylation. Let us see this in NCERT also. See. When only PS1 is functional, the electron is circulated within the photosystem and the photophosphorylation occurs due to cyclic flow of electrons. A possible location where this could be happening is the stroma lamellae, while the membrane of lamellae of the grana have both PS second and stroma lamellae. Membrane lack PS second as well as, See, while the membrane of lamellae of the grana have both PS1 and PS2, the stroma lamellae membrane lack PS2 as well as NADP reductase enzyme. This NADP reductase enzyme, in short form, it is called as FNR enzyme. The excited electron does not pass on to NADP plus, but is cycled back to PS1 complex through the electron transport chain. This point, remember this point I was telling you. The cyclic photophosphorylation results in the synthesis of ATP but not NADPH. Cyclic photophosphorylation also occurs when only light of wavelength beyond 680 nm are available for excitation. Why? Because beyond 680 nm, PS2 cannot function. The maximum functioning of the PS2 is 680 because the reaction center can absorb light of maximum 680 nm. So this we have completed the electron transport system and the photophosphorylation. Is it clear, children? Just give me a quick review so that I can move forward. Now, children, the question is, we said that due to this electron flow or this electron flow, there is synthesis of ATP. But what is the actual mechanism of ATP synthesis? Till now we said that due to downhill journey of electron, ATP is synthesized. This is photophosphorylation. But what is the actual mechanism? This actual mechanism of ATP synthesis was discovered by a British scientist, Peter Mitchell. And in his own language, he called this as the chemiosmotic hypothesis. Although now it is proved that this is the method, but still the term hypothesis is continuing. It should now be called as theory, but still we call it as hypothesis. So let us see the actual mechanism of ATP synthesis. As I said, it is called as chemiosmotic hypothesis. It was given by a British scientist, Peter Mitchell. What Peter Mitchell said, that when there is electron transport in a membrane, due to this electron transport, protons move from one side of the membrane to the other side. Okay, so due to electron transport, there is a proton transport from one side of the membrane to the other side of membrane, proton pumping we can rather say. Due to this, a proton gradient is created across the membrane. And when this proton gradient is broken down, sufficient energy becomes available for the synthesis of ATP. This is the basic of this, fine. Now, how electron transport results in creation of a proton gradient, let us try to see here. There are three reasons for this. So we are going to discuss the chemiosmotic hypothesis. First of all, three reasons I said for creation of proton gradient across the thylakoid membrane. First of all, we have said that at PS2, for continuous supply of electron to the electron transport chain, at PS2 there is splitting of water, light-induced splitting of water. Due to this, the protons that are released from water accumulate here. It means accumulate here. It means toward the lumen side of the membrane, there will be more protons as compared to the stroma side. So this is the first reason of creation of proton gradient. Second, when the primary acceptor of PS2 gives its electron to plastoquinone, remember plastoquinone is not simply an electron transporter, it is a complete hydrogen transporter. Complete hydrogen transporter means it is transporter of both electron and proton. So when plastoquinone takes electrons from the PS2, it also pulls protons from the stroma. And through the cytochrome B6F complex, it pumps these protons into the pumping I am calling because it is requiring energy. Where is the energy coming from? Energy is coming from the downhill process of the electron. Remember, when something will move from the higher energy to lower energy level, energy will become released, become available for any function to be performed. So since electrons are moving downhill, so the energy which is released due to downhill movement of electron, it helps the plastoquinone to pump protons from the stroma into the thylakoid lumen through cytochrome B6 complex. It means again there is increase of protons in the lumen side and decrease of protons to the stroma side. So second reason for creation of proton gradient. And third reason, when this NADP is reduced to NADPH, protons are taken from the stroma. So further decrease in the amount of protons in the stroma. So this is the third reason. As a result, a strong electrochemical potential gradient is created. So very high protons in the lumen side and very less protons in the stroma side. Now, you know a basic property of all ions or molecules is diffusion. That is due to their own kinetic energy, their movement from the region of their higher concentration to the region of their lower concentration. So these protons will also have the tendency to diffuse out, because here there is a high concentration of protons and here outside low. But the membrane is not freely permeable to charge. This you have studied in cell biology. The cell membrane, the lipid bilayer of the cell membrane does not allow charged particles to pass through it. It can allow only the neutral solutes to pass through it. But in the membrane, there is a tunnel protein, a channel protein called as CF0. This channel protein provides facilitated diffusion to the protons, so back into the stroma. When the CF0 provides facilitated diffusion to the protons from the lumen back to the stroma, there is breakdown of the proton gradient. Okay, this breakdown of the proton gradient releases sufficient energy, and this energy is used by CF1. I will slightly enlarge it, you can understand. See here. I think now it is visible. See. So when there is breakdown of the proton gradient through the CF0, sufficient energy is released, and attached to the CF0 is a peripheral protein CF1, which you see has ATP synthase activity. So this energy is used by the ATP synthase or F1 to produce ATP from ADP. It means the ultimate reason of ATP synthesis is the breakdown of proton gradient. But for the breakdown of proton gradient, first should be creation of proton gradient. For the creation of proton gradient, there should be electron transport, because only electron transport can create the proton gradient. And for the electron transport, there should be excitation of the chlorophyll. And for excitation of the chlorophyll, there should be light absorption. So light absorption, chlorophyll excitation, electron transport system, creation of proton gradient, breakdown of proton gradient, and synthesis of ATP. Is this point clear, everyone? Because this is one point where you people get stuck. That is why I'm asking. Is it clear, absolutely clear to everyone? Hello? Fine, okay, fine. So this is all. So it means what are the requirements of the chemiosmotic hypothesis? Let us see the requirements of chemiosmotic hypothesis. So requirement of chemi-hypothesis, first of all, there should be a membrane across which proton gradient should be created. Second, for creation of proton gradient, there should be pumping of protons from stroma into the thylakoid lumen. Third, due to this creation of a proton gradient, and when there is breakdown of proton gradient, there should ATP synthase will produce ATP. So 4 requirements for chemiosmotic hypothesis: a membrane, a proton pumping, a proton gradient, and an ATP synthase. So with this children, we complete the light reaction of photosynthesis. So if we see the products of light reaction of photosynthesis, ATP, NADPH plus H plus, and O2. O2 is released into the atmosphere as the final product, while this ATP and NADPH are immediately used in the stroma in the dark reaction to produce glucose. Okay, so that is why if we switch off the light, for some time, the dark reaction will continue since there is surplus ATP and NADPH, and after that, when ATP and NADPH are over, dark reaction will also stop. So let us see. Where are the ATP and NADPH used? We learned that the products of light reaction are ATP, NADPH, and O2. This question is very frequently asked. Why I am saying so? Because you remember ATP and NADPH, but you forget O2. So remember, O2 is also a product of light reaction, although it is not used in the dark reaction. But all 3 are products of light reaction. ATP and NADPH. Of these, O2 diffuses out from the chloroplast, while ATP and NADPH are used to drive process leading to synthesis of food, more accurately the sugar. This is the biosynthetic phase. Why we are calling it biosynthetic phase? Because it is enzyme catalyzed multi-step process. That is why we are calling it as biosynthetic phase. Fine. This process does not directly depend on the presence of light, but is dependent on the products of light reaction, that is ATP and NADPH, besides CO2. This is also a very important question. That what are the requirements for the dark reaction? So the requirements of dark reaction are this CO2 and H2O, because H2O will release hydrogen to reduce CO2 to produce glucose, and to carry out this process energy source will be required, which are ATP and NADPH. Okay, fine. So all are the dark reaction products. You may wonder how this could be verified. It is simple. Immediately after light becomes unavailable, the biosynthetic process continues for some time and then stops. Why? Because now, since light reaction is stopped, production of ATP and NADPH has stopped. If then light is made available, the synthesis starts again. Can we hence say that calling the biosynthetic phase as dark reaction is a misnomer? Tell me. See how many people answer. Is calling dark reaction a misnomer? Wrong naming? Because if we say dark reaction, it means it can independently take place in dark. Now coming to dark reaction or biosynthetic phase. Site stroma. Two types of cycles of dark reaction were discovered. First was discovered by Melvin Calvin, so that is why called as Calvin cycle. And first product is a 3-carbon compound phosphoglyceric acid, that is why it is also called as C3 cycle. Most of the plant, it is seen in all plants. Okay. Now, later on, one more cycle was discovered, which is called as Hatch and Slack cycle or C4 cycle, because the first product is a 4-carbon compound. Someone is saying, what can you, sir, can you tell what is that misnomer? Misnomer means giving a wrong name. Okay, giving a wrong name to someone, you are giving a wrong name, that is called as misnomer. So do you think that the name dark reaction given is a wrong name? It is just like if you call potato a green vegetable, it is a misnomer. Understood? Fine. So now Calvin cycle is present in all plants because Calvin cycle is the only cycle through which sugar can be synthesized. So on this basis, there are two types of plants. C3 plants that show only Calvin cycle or C3 cycle, only C3 cycle. And C4 plants show both C3 and C4 cycle. So C4 plant both C3 and C4 cycle, because the ultimately sugar is synthesized through the Calvin cycle. So first of all, let us see the Calvin cycle. It was discovered by Calvin and Benson. Calvin and Benson discovered, that is why it is called as the C3 cycle. They used radioactive carbon C14 in CO2. So see where this carbon is going, because if a compound is showing radioactivity, its presence can be easily traced out by a process called as radio isotopic tracer technique. By this technique, we can discover where this radioactive carbon is present, okay, and they used chlorella, a green alga, for their experiment. All these questions are asked. Okay, so they used the technique, radioisotopic tracer technique. They used radioisotopic tracer technique, and they used CO2 which had radioactive carbon C14, and the experimental organism were chlorella, green alga. Okay, fine, which is the unicellular green alga chlorella. Fine. CO2 acceptor is a 5-carbon compound, RUBP, ribulose bisphosphate. Okay, and first stable product is a 3-carbon compound phosphoglyceric acid. And since first stable product is a 3-carbon compound phosphoglyceric acid, that is why it is called as the C3 cycle. Cycle operates 6 times to produce 1 glucose molecule, because 1 cycle will use 1 molecule of glucose, and you know 1 cycle will use 1 molecule of CO2. I'm sorry, 1 cycle will use 1 molecule of CO2. Okay, and for synthesizing 1 glucose, 6 CO2 will be required. That is why cycle operates 6 times to produce 1 molecule of glucose. Fine. And the cycle has 3 phases. Let us try to see the 3 phases of the cycle. See. First phase is carboxylation. So carboxylation, adding CO2 to any compound, is called as carboxylation. So the 5-carbon compound RUBP is carboxylated. Water is also used to produce 3-phosphoglycerate. Okay, 3-phosphoglycerate is a 3-carbon compound. That is why we are calling it a 3. Now, this 3-phosphoglycerate uses the products of light reaction ATP and NADPH to produce triose. Okay, to produce triose. Some triose molecules are used for synthesis of glucose and ultimately starch. From triose, first glucose will be produced, and from this glucose ultimately starch will be produced. While remember, it is a cyclic process, so it will continuously require RUBP. So the RUBP that you have used here, you have to regenerate the RUBP. So some triose are used for synthesis of glucose, remaining triose are used for the regeneration of the RUBP. So that is why the cycle has 3 phases. First, carboxylation to produce PGA, then reduction to produce triose, some triose used for glucose synthesis, remaining triose in the 3rd phase to regenerate the RUBP, and this process keeps on continuing. And you see, for production of 1 glucose molecule, 6 molecules of CO2 will be used, 18 molecules of ATP will be used, and 12 molecules of NADPH. So 6 CO2 molecules, 18 ATP and 12 NADPH molecules are used to synthesize 1 glucose molecule. Now if you see, there is a disparity in the amount of ATP and NADPH used. So if only non-cyclic photophosphorylation operates, then you will get 12 ATP and 12 NADPH. But it is requiring 6 additional ATP. So this, where does this 6 additional ATP comes? This 6 additional ATP comes from the cyclic photophosphorylation. That is why both non-cyclic and cyclic photophosphorylation operate. So 12 ATP and 12 NADPH come from the non-cyclic photophosphorylation, and additional 6 ATP come from the cyclic photophosphorylation. Let us see this also. See, so So for 6 CO2, 18 ATP and 12 NADH, it means for 1 CO2, how many ATP? 3 ATP and 2 NADPH. Fine. So see, hence, for every CO2 molecule entering Calvin cycle, 3 molecules of ATP and 2 NADPH are required. This point, see, it is probably to meet the difference in the number of ATP and NADPH used in dark reaction that the cyclic photophosphorylation takes place. To fulfill this extra requirement of ATP, cyclic photophosphorylation takes place. Fine. Okay. So this is all regarding the Calvin cycle or C4 cycle, C3 cycle. Next we come to C4 cycle. C4 cycle, there are certain plants which grow in dry tropical area. Okay, there are certain plants which grow in dry tropical area. In these plants, the first product of the dark reaction was not found to be a 3-carbon compound phosphoglyceric acid, but it was found to be a 4-carbon compound oxaloacetic acid. Since first product was a 4-carbon compound oxaloacetic acid, it came to be known as the C4 cycle. Okay. In these plants, there are certain specific features. First of all, in these plants, along with mesophyll cell, bundle sheath cells also contain chloroplast. The bundle sheath around the vascular bundle of leaf also contains chloroplast. And there are many concentric rings of bundle sheath cells around the vascular bundle, and this type of anatomy is called as Krebs anatomy. Krebs means border-like, bordering, wreath-like. So since multiple bundle sheath cells, multiple rings of bundle sheath cells are present around the vascular bundle, that is why it is called as Krebs anatomy. Second thing is, bundle sheath cell contain large number of large size chloroplasts. This is also one of the reasons for high productivity of the C4 plants. Fine. So this is the second thing. Third, bundle sheath cells have no intercellular spaces. They are very tightly packed and have thick impermeable walls. So mesophyll cells also have chloroplast, bundle sheath cells also have chloroplast. That is why in these plants, CO2 is fixed twice. First in the mesophyll cell and second in the bundle sheath cell. How it takes place, let us see. See here. A small water break. Is taken and the CO2 acceptor is a 3-carbon compound phosphoenol pyruvate, which is a 3-carbon compound. It is the CO2 acceptor, and this forms a C4 acid called as oxaloacetic acid. So 3-carbon plus 1 4-carbon compound, and this oxalo acid is then reduced to malic acid. Malic acid is again a 4-carbon acid. Fine. So this is called as fixation. So CO2 is fixed with the help of 3-carbon compound PEP, and the enzyme that catalyzes this process is called as the PEP carboxylase. So in the presence of enzyme PEP carboxylase, someone is asking, sir, is it required to read photorespiration? Everything is important, beta. Questions from photorespiration also come. Because you see, NCERT is itself a concise thing, so nothing we can leave. We will see how much we can cover, that we will see. Fine. Okay. So this 4-carbon acid, malic acid, is not transported to the bundle sheath cell. In bundle sheath cell, there is decarboxylation of the malic acid to produce a 3-carbon acid and CO2. Now this CO2 which is released in the bundle sheath cell, here it takes place Calvin cycle. I told you that Calvin cycle is present in all plants. In C3 plants, it takes place in the mesophyll cell. In C4 plant, it takes place in the bundle sheath cell. So as a result, there is fixation of the CO2 to produce sugar in the bundle sheath cell. Now the C3 acid, it returns back, and this C3 acid regenerates the phosphoenol pyruvate. Fine. So if we see, it is something like this. Mesophyll cell, PEP 3 carbon plus CO2 1 carbon. Peptase or PEP carboxylase enzyme, oxaloacetate 4C acid. This 4C acid is reduced to another 4C acid, malic acid, another 4C acid. Okay. Transported to bundle sheath. In some plants, instead of malic acid, aspartic acid is also transported. So this is the mesophyll cell. Okay. So this is called as fixation. And this transportation I already told you. Next, in bundle sheath cell, malic acid 4 carbon, decarboxylation. So it forms pyruvic acid 3 carbon compound plus CO2. CO2 for fixation in Calvin cycle to produce glucose. Fine. And this pyruvic acid transported back to mesophyll cell where it regenerates the phosphoenol pyruvate. Fine. So that is why in these plants CO2 is fixed twice. Primary CO2 fixation in mesophyll cell and secondary CO2 fixation in the bundle sheath cell. And ultimately glucose is synthesized in the bundle sheath cell. While in C3 plants, photosynthesis takes place only in mesophyll cell, so the glucose is synthesized in the mesophyll cell. Fine. This chart is important. We will do this chart and that will be the end of this chapter. Before that, one small phenomena, photorespiration. Photorespiration is, children, a process that takes place only in C3 plants. Second, why we are calling it as respiration? Because it releases CO2. Why photo? Because it takes place only in presence
Sir. Sir, I changed it, sir. I changed it.
Hello, is it audible now? Can you hear me now? Okay, fine. So I'm saying that enzyme Rubisco has dual nature. Okay, if normal conditions, then it causes carboxylation of the RUBP, and this is Calvin cycle, you know, first step. But same enzyme Rubisco, if there is high O2 is to CO2 ratio, instead of binding CO2, it binds O2 to the RUBP. This is called as oxygenation. And this is the product. These questions, all these questions are asked in exam. Okay. Then this leads to the process of photorespiration. Why this happens? Because children, the Rubisco enzyme has common site for binding of O2 and CO2. Okay, common site for binding of O2 and CO2. So this is a competitive binding. Either O2 will bind or CO2 will bind. Remember one thing. Atmosphere has very less CO2 as compared to oxygen. But still, Rubisco prefers binding to CO2. It means Rubisco has more affinity for CO2. This question was asked in NEET 2025. There was a question. Forget. What is wrong about Rubisco? There were 4 options. Fine. So Rubisco has more affinity for CO2 despite atmosphere having very little CO2 as compared to O2. It prefers binding with CO2. But when this O2 concentration becomes even higher, when these gaps become even wider, then some, of course, some O2 does bind to the Rubisco, and then the process of photorespiration starts. So the 3-4 important points. First of all, it is seen only in C3 plants, not in C4 plants. Second, it starts in chloroplast, but it involves 3 organelles: chloroplast, peroxisome, and mitochondria. Third, the white we are calling for respiration because CO2 is released from the mitochondria. It is not a useful process. You see, photosynthesis is a useful process because it gives glucose. Respiration is a useful process because it gives ATP. Photorespiration does not give any glucose or any ATP or any NADPH. It is a wasteful process because rather it uses ATP. And it causes loss of CO2, which CO2 that would have gone for photosynthesis that is lost in the atmosphere. So it is a completely wasteful process. So this is all regarding the photorespiration. Fine? Okay. Next, last we are coming to this chart. Very, very important chart. C3 plants, C4 plants. Cell type in which Calvin cycle takes place. So in C3 plants, Calvin cycle takes place in mesophyll cells, while in C4 plant, Calvin cycle takes place in the bundle sheath cell. Cell type in which the initial carboxylation reaction occurs. Of course, initial carboxylation in both will occur in the mesophyll cell. But in C3 plants, initial carboxylation by RUBP, and C4 plants, initial carboxylation by the pepcase. Third, how many cell types does the leaf have that fix CO2? C3 plants have only one type, okay, that is the mesophyll cell. C4 plants have 2 types, mesophyll cell and bundle sheath cell. Fine. Which is the primary CO2 acceptor? In C3 plant, the primary CO2 acceptor is a 5-carbon compound RUBP. In C4 plant, the primary CO2 acceptor is a 3-carbon compound phosphoenol pyruvate. Number of carbons in the primary CO2 acceptor, already I told you 5 carbon and 3 carbon. Which is the primary CO2 fixation product? In C3 plants, it is the phosphoglyceric acid, and C4 plants, it is the oxaloacetic acid. Next, number of carbons in primary CO2 fixation product. C3 plants, it is 3 carbon, and C4 plant, it is 4 carbon. Does plant have Rubisco? Yes, Rubisco is present in all plants because Calvin cycle is present. Does plant have pepcase? Pepcase is present in C4 plants, not in C3 plants. Fine. Which cell in the plant have rubisco? In C3 plants, rubisco is present in the mesophyll cell. In C4 plants, rubisco is present in the bundle sheath cell. CO2 fixation rate under high light condition. The C3 plants are not adapted to very high light. So in C3 plants, under high light condition, due to photooxidation of chlorophyll, photosynthetic rate will decrease. While C4 plants are adapted to high light intensity. So they will respond to high light intensity by high rate of photosynthesis. Whether photorespiration is present in low light intensity? No. For photorespiration, three conditions should be fulfilled. First, high O2 is to CO2 ratio. Second, high temperature. And third, high light intensity. So if light intensity is low, there will be no photorespiration even in C3 plants. Whether photorespiration present at high light intensity? Yes. In C3 plants, high light intensity will promote photorespiration. But C4 plants, I told you, in any condition do not show photorespiration. Whether photorespiration would be present at low CO2 concentration? Yes, because it is O2 is to CO2 ratio. So if CO2 concentration is low, then C3 plants will show photorespiration, but as I said, C4 plant do not show photorespiration. Whether photorespiration would be present at high? No. If CO2 concentration is high, rubisco will always bind to CO2. There will be no photorespiration either in C3 plant nor in C4 plants. Temperature optimum. C3 plants are adapted for moderate, that is 20 to 25 degrees centigrade, while C4 plants above 40 degrees centigrade. Okay. Examples: most of the plants on earth are C3 plants. That is wheat, rice, tomato, potato, all are C3 plants. C4 plants example are rare. You can remember three examples: sugarcane, maize, and sorghum. Remember three examples: sugarcane, maize, and sorghum. These are the examples of the C4 plants. So children, with this we are completing the chapter of photosynthesis. Fine. Any specific question you have in chapter of photosynthesis, you can ask. Hello. Law of limiting factor. Someone has asked law of limiting factor. You see, photosynthesis is a process that is controlled by many factors: temperature, light, CO2, many factors. So if any process is controlled by many factors, then the rate of process is controlled by that factor which is present in its minimum amount, or the rate of process is limited by the factor that is present nearest to its minimum value. And any increase in the value of this factor causes increase in the rate of process. This is called as law of limiting factor. So if a process is conditioned to its maximum velocity by a number of multiple factors, then the rate of the process is controlled by the value of that factor that is nearest to its minimum value. Okay, or the rate of process is limited by the value of that factor which is nearest to its minimum value. Any increase in the value of this factor causes in the rate of the process. Okay, and this is the major factor which we call it. For example, C3 plants, their optimum CO2 requirement is more than 450 microliter per liter. But the atmospheric CO2 concentration at present is 360 microliter per liter. It means for C3 plants, the most important limiting factor every day is the CO2. Because the requirement is more than 450, atmosphere is giving only 360. So CO2 is a major limiting factor for C3 plants every day in the atmosphere. Thanuj Kumar, did you understand now? Achha, graph of light intensity. Okay, I'll explain. No problem. Graph of light intensity, I will explain. See. Someone is saying, explain the graph of light intensity for factors. See. See, When light intensity is low, with increase in light intensity, there is also increase in the rate of photosynthesis. It means light is the limiting factor. Light is the limiting factor. Above all other factors, the minimum is of light. So as we increase the light intensity, rate of photosynthesis is increasing. This is called as a linear relationship. So if we see here, from here to here, if we see A is light is the limiting factor, fine. But with further increase in light intensity, there is no further increase in the rate of photosynthesis. Means now sufficient light is present, light is not the limiting factor. So if we see this C, it means light is no more the limiting factor. Some other factor, and most common, I told you, CO2 is the limiting factor. So at low light intensity, when light is a limiting factor, there is a linear relationship between the light intensity and the rate of photosynthesis. With increase in light intensity, rate of photosynthesis increases. But a time comes when the light is sufficient. With further increase in light intensity, there is no increase in the rate of photosynthesis, because now light is no more the limiting factor. It means from here to here, there is transition from one limiting factor to another limiting factor. So A, light is the limiting factor. C is light is no more the limiting factor. Some other factor is limiting factor. And B is a transition from one limiting factor to another limiting factor. Now, if you come to here, this is D. It means this is the optimum light intensity, because at this light intensity, maximum photosynthesis is taking place. So D is the optimum light intensity or light saturation point. For most of the plant, it is just 10% of the full sunlight. It means generally, if it is a sunny day, means cloud is not there, and if the plant is not very deep inside the forest, light generally does not function as a limiting factor. Gyan Prakash, beta, is it clear now? Any other question you want to ask? Gyan Prakash? Okay, fine. Thank you. Respiration in plants. Basically, we are discussing here about cellular respiration. Children, you see that within 4 or 5 hours, we have to complete at 7 or 8 chapters. So naturally, touching every point will not be possible. Okay, touching every point will not be possible. That is why I'm mostly focusing on those points from where maximum questions are asked. So that is why if I'm leaving certain point, in that point, if there is something you think that you are not clear with, you ask, I will tell you. But you can also understand the practical problem that every point could not be touched in such short span of time, right? I'm trying to say touch the hot spots, okay? And to maintain a continuity of the topic, so there is no break in the continuity of the topic. That is the only thing, right? Okay. Okay, can we continue? Fine. So, cellular respiration is enzyme controlled multi-step process where a compound, you can say food, is oxidized and energy is released. Major part of this energy is lost as heat, and remaining part is conserved as high energy molecule ATP. And when we perform any work, we use this energy of ATP. That is why ATP is called as energy currency of cell. I am talking to you, I am using ATP. If you are writing, you are using ATP. But if you are listening, by thinking also, then you are using ATP, and but you are only listening, then you are not using any ATP. So it depends whether you are using your ATP or not, but I am using my ATP, I am very clear about it. Is it clear? Haji. As I said, it is a multi-step process. So what is the advantage of multi-step process? First of all, if a glucose molecule is completely oxidized in one step, so much energy will be released that it will burn the tissue. But when it is released in small steps, the temperature of the tissue does not increase. Second, if it is a multi-step process, Ajay, you are using UATP, very good, very good. Okay, fine. So, if it is a multi-step process, first, the temperature of the tissue does not increase. And second, due to multi-step process, suppose glucose is converted into A, into B, B into C, C into D. It means many intermediate compounds are produced. These intermediate compounds are called as the carbon skeleton. So due to multi-step process, many carbon skeleton are produced, and these carbon skeleton, many of these are used for synthesis of other compounds. That is why we say that respiration is amphibolic process. Amphibolic process. Why? Because breakdown of glucose into CO2 and water. This is catabolism. But the carbon skeleton or intermediate compounds is used for synthesis of other compounds. For example, if you are breaking down a glucose, a point you will get acetyl-CoA. This acetyl-CoA can be used for synthesis of carotenoids. It can be used for the synthesis of fatty acids. This is anabolism. It means respiration process is involved in both catabolism and anabolism. That is why it is called as amphibolic process. Fine? Okay. Now, types of respiration. Aerobic respiration. So, here oxygen is used, that is why it is called aerobic respiration. And due to use of oxygen, the respiratory substrate is completely broken down. How will you come to know complete breakdown? Because in the end, you are not getting any organic compound. So if the end you are not getting any organic compound, it means there is no organic end product. It means all the carbon-carbon bond have been broken. Remember, during respiration, the carbon-carbon bond is broken, and due to this, energy is released, and this energy is used for synthesis of ATP. So since there is no organic end product, it means all carbon-carbon bonds have been broken. So more carbon-carbon bonds will be broken, more energy will be released. That is why it gives a very large amount of energy, aerobic respiration. Second is anaerobic respiration, which we commonly call as fermentation. Here O2 is not used. Okay. O2 is not used. That is why all the carbon-carbon bonds are not broken. There is partial breakdown of the respiratory substrate. How you can see partial breakdown? Because in the end also you are getting a organic compound. So organic end product means still some carbon-carbon bonds are there which have not been broken. So due to this, since all carbon-carbon bonds have not been broken, that is why very less amounts of energy is released. Only less than 7% of the total energy present in glucose is released. So you can see 7% of 6A. Total energy released this much. Here only less than 7%. So 58, 59 kilocalorie energy only released in this process. So this process is highly inefficient process. Then why does it take place? Remember children, when life originated on the earth, the atmosphere of the earth was anaerobic. There was no oxygen on earth. So earlier organisms, all that came on the earth, they were all performing only fermentation because no oxygen available. Later on, when cyanobacteria came, Cyanobacteria for the first time used water in their photosynthesis. So due to this, they start releasing oxygen. Then the atmosphere of the earth became aerobic. Then aerobic organisms evolved. So initially, all the organisms on earth in the initial when life originated were all only anaerobics or fermentation. Even today, many of them are still performing only fermentation. For example, methanogens. Methanogens are obligate anaerobes. Means if you give oxygen, they will die. On the other hand, certain organisms are facultative anaerobe. Facultative anaerobe means basically they are aerobic. But in the absence of oxygen, they will not die. In the absence of oxygen, they will switch over to fermentation. These are called as facultative anaerobes. And yeast is a very good example of facultative anaerobe. Fine. So remember that methanogens are obligate anaerobes. They cannot survive in presence of oxygen. Yeast is facultative anaerobe. It is basically aerobe, but in the absence of oxygen, it will switch over to fermentation or anaerobic respiration. Fine. Okay. So this is all regarding now. If we see the steps in cellular respiration. So first is glycolysis. Glycolysis. It was discovered by three people, Engelmann, Meyerhoff and Parenes. That is why it is also called as EMP pathway. Embedded Meyerhoff and Parenes, that is why it is also called as EMP pathway. It takes place in cytoplasm, found in all organisms. Means common to both aerobic respiration as well as fermentation. Okay, it is found everywhere. Okay, partial breakdown of glucose, a 6-carbon compound, to form 2 molecules of pyruvic acid, which is a 3-carbon compound. Net ATP gain, 2 ATP. This question is very commonly asked. What is the net gain of ATP from glycolysis? So you have to get 4 ATP are produced, 2 are used. That is why net gain is 2 ATP. Fine. Okay. So this is it is a linear process. This is not a cyclic process. So let us see it here. See, it is a 10-step process. First of all, there is phosphorylation of glucose. Glucose, which is a 6-carbon compound, first it uses a one molecule of ATP to convert into glucose 6-phosphate. So initially there is use of ATP. Okay, glucose 6-phosphate. Then glucose 6-phosphate undergoes isomerization. So first step is phosphorylation of glucose. Second is isomerization. So glucose 6-phosphate is isomerized to fructose 6-phosphate. Third step is again, the enzyme of first step is hexokinase. This is important. This question was also asked 2 years back. So the enzyme for first step is hexokinase. For second step, enzyme is isomerase. And third step is again phosphorylation of fructose. So fructose 6-phosphate is converted into fructose 1,6-bisphosphate. Means 1 phosphate attached also on the first carbon. This is catalyzed by the enzyme. Okay. So this is the very, very important step because it is the rate limiting step of the glycolysis. It is the rate limiting step of the, and the enzyme is called as phosphofructokinase. The enzyme that catalyzes conversion of fructose 6-phosphate into fructose 1,6-bisphosphate is called as the phosphofructokinase PFK. And this enzyme is the main rate controlling step of glycolysis. That is why this enzyme is also called as the pacemaker enzyme of glycolysis. Fine. Now fructose 1,6-phosphate now splits into 2 triose. Remember, 6-carbon sugars are called as hexose sugar. 3-carbon sugars are called as triose sugar. So it splits into 2 molecules of 3-carbon sugars or triose sugar. One is called as phosphoglyceraldehyde and one is called as dihydroxyacetone phosphate. Fine. PGL and DHAP. And they are interconvertible. They are isomers. They are interconvertible. Now the triose undergoes oxidation. Triose undergoes oxidation to convert into 1,3-dipga. So from phosphoglyceraldehyde we get 1,3-dipga, and due to oxidation the hydrogen that is released, that is taken by NAD to it produces NADH. So there is production of NADH. This is oxidation step. Okay, and simultaneously also one inorganic phosphate is attached. So this step involves two things. First of all, oxidation of the PGL into 3-PGA and also phosphorylation. That is why you see it is converting into 1,3-bis-phosphoglyceric acid. Next step is the ATP gaining step. 1,3-bis-PGA is converted into 3-PGA. As a result, 1 ATP is produced. So when ATP is directly produced from the substrate, this is called as substrate level phosphorylation, or in short form it is called as SLP. So this is the first step where we start getting ATP. Okay, so this is called as substrate level phosphorylation. Now 3-phosphoglyceric acid undergoes isomerization. The phosphate group shifts from third carbon to second carbon, so we get 2-PGA. Okay, from 3-PGA to 2-PGA. Now 2-PGA undergoes dehydration, loss of water. So it converts into phosphoenol pyruvate. Okay, a 3-carbon compound, phosphoenol pyruvate. And phosphoenol pyruvate undergoes again substrate level phosphorylation to produce pyruvate. So again there is gain of ATP. So there are one step where there is oxidation or dehydrogenation, NADH production, and 2 steps substrate level phosphorylation. This step and this step. Fine. Now remember, you started with your process with a 6-carbon compound glucose, and from here you are moving to 3-carbon compound. And this will also be converted into, this PGL will also, DHAP will also convert into PGL. It means you will get 2 molecules of PGL. That is why from here onwards, the entire process will be multiplied by 2, because 1 PGL this and 1 PGL due to isomerization of the DHAP. So from here, so it means here you are getting 2 ATP, here you are getting 2 NADH plus H plus, and again here you are getting 2 ATP. So total ATP gain is 4 ATP. But in the beginning already you have used 2 ATP, 1 ATP here and 1 ATP here. That is why the net gain is only 2 ATP. So from glycolysis, net gain from glycolysis is 2 pyruvic acid, 2 ATP and 2 NADH. Okay, so this is the product of glycolysis: 2 pyruvic acid, 2 ATP and 2 NADH. 4 ATP produced, 2 used, that is why net gain is only 2 ATP. Fine. Okay, so what will happen to this pyruvic acid? Now what will be the future of this pyruvic acid? It depends upon the availability of oxygen and the requirement of the cell. Okay, if oxygen is not available, it will undergo fermentation. If the requirement of ATP becomes very high, so that the available O2 is not able to fulfill that requirement, then also it will start fermentation. And if oxygen is available, then it will go for aerobic respiration. So pyruvic acid can undergo fermentation or it can undergo aerobic respiration depending upon 2 things I said. First, availability of oxygen, and second, demand of the cell. So first of all, we are going to see the fermentation. Let us see. Fermentation. See here. Up till glycolysis it is common. Okay, so up till pyruvic acid it is common. From pyruvic acid, either this pyruvic acid can undergo alcoholic fermentation to produce ethanol and CO2, or this pyruvic acid can undergo lactic acid fermentation. In lactic acid fermentation, CO2 is not produced. Okay, whether it is alcoholic fermentation or lactic acid fermentation, during the conversion of pyruvic acid to ethanol or lactic acid, there is no ATP production. So that is why in fermentation, the total ATP production is only 2 ATP that is produced in the glycolysis. After glycolysis, there is no further oxidation step, so there is no further ATP production. So per glucose, if you are asked net ATP gain from fermentation, it will be only 2 ATP. Whether it is alcoholic fermentation or it is lactic acid fermentation. Let us see these two fermentations. Alcoholic fermentation. Pyruvic acid, pyruvate, T carboxylase. Okay, so acetaldehyde plus CO2. Since CO2 is released, it is called as decarboxylase. And second, acetaldehyde plus NADH. This is the same NADH that was produced in the glycolysis. Okay, NADH. Alcohol dehydrogenase. This will produce ethanol plus NAD plus. So NAD is oxidized. Fine. So it means the product of alcoholic fermentation are two things: CO2 and ethanol. Fine. That is why, and it is performed by organisms like yeast. And you know, ethanol is used as alcoholic drink or alcoholic beverage, ethyl alcohol. That is why in breweries, yeast is used for the production of ethanol. But children, what happens when the alcohol percentage reaches 13%? This ethanol is poisonous even to the yeast that is producing it. So, or toxic to the yeast. So when the alcohol percentage reaches 13%, the yeast poisons itself to death due to this alcohol. That is why an alcoholic drink that is produced only by fermentation, it has maximum 13% alcohol like wine and beer. Now, if you want to further increase the alcohol percentage, you will have to remove water. That is, you have to undergo distillation. So by distillation, you can increase alcohol percentage up to 100%. Okay. So that is why the alcoholic drinks that have 40% or more alcohol in them, first fermentation is done, then distillation is done. That is why they are called as distilled alcoholic beverages like rum, whiskey, brandy, gin. So rum, whiskey, brandy and gin are the distilled alcoholic beverages because after fermentation there is distillation. And beer and wine are non-distilled alcoholic beverages because they are produced only by fermentation, not by distillation. So that is why alcohol percentage is 13% or less. Is it clear to everyone? Next. Lactic acid fermentation. This is caused by lactic acid bacteria, lactobacillus, etc., which are used for the production of curd. Apart from that, are you there? No response? Hello? Are you getting the points? Okay, fine. Fine. Actually, you all became so silent, I thought. Up till alcoholic fermentation, is it clear to everyone? Up till alcoholic fermentation, is it clear to everyone? Okay, fine. Now, it is performed by lactic acid bacteria, lactobacillus, which are used for the production of curd, perugu. Fine. It also takes place in our skeletal muscles when we are doing very rigorous exercise, very strenuous exercise. You go in gym, na? In gym, a lot of exercise people perform. So what happens? The requirement of ATP is so high that the available oxygen is not able to fulfill that demand. So then to supply rapid ATP, then fermentation also starts. Okay. So in our skeletal muscles also it takes place when we are performing very rigorous exercise. This is a single step process, pyruvic acid. plus NADH. This is NADH that was produced in the glycolysis. Lactic acid dehydrogenase. As I said, during lactic acid fermentation, there is no CO2 production. So CO2 is not produced in this process. So this is all. As I told, whether it is lactic acid fermentation or alcoholic fermentation, there is only net gain of 2 ATP. Less than 7% energy present in this. This question was asked last year. Less than 2024. Less than 7% energy present in the glucose is released, and not all of this energy is used in ATP production. So very few energy is released, and that also maximum part is lost. So very few of that 7% is also used in ATP synthesis. Fine. So this is all regarding fermentation. It means the fermentation is a very less efficient process, but sometimes it becomes there is no other option. If oxygen is not available, if ATP demand is very high, then this process takes place. Fine. Now coming to aerobic respiration. The site for aerobic respiration in eukaryotes is mitochondria. Okay. Let us see what happens in aerobic respiration. Just a minute. These 2 points are very important, children. Very important points if you can understand. First, the complete oxidation of pyruvate. Pyruvate was the end product of glycolysis. In aerobic respiration, it is completely broken down. Means all carbon-carbon bonds are broken by the stepwise removal of all the hydrogen atom. Okay. So from the pyruvate, all hydrogen atom will be removed. Okay. So what will be left? CO2. So from the pyruvate, when all hydrogen are removed, CO2 is left, and this CO2 is released in the atmosphere. That is why in respiration CO2 is released. Now this H which is released during this process, this splits into 2 protons and 2 electrons, and these electrons, they pass on the electron removed as a part of the hydrogen atom of molecule O2 with simultaneous synthesis of ATP. So this electron is ultimately taken by O2 to produce water. Okay, fine. And during this process, there is electron transport leading to ATP synthesis, same as you saw just sometimes back in the chloroplast. So whenever on the membrane there is electron transport, there is creation of proton gradient, and breakdown of proton gradient leads to ATP synthesis. In chloroplast, this electrons were moving on thylakoid membrane, and in mitochondria, these electrons move on the inner mitochondrial membrane. So the process is similar. The only thing is site. So here, these electrons move on a chain of electron carriers present in the inner mitochondrial membrane. There, the electron was ultimately accepted by NADP. Here, the electron is ultimately accepted by oxygen. Okay. But electron transport creates a proton gradient, and breakdown of proton gradient produces ATP. Same chemiosmotic hypothesis here also applies. Fine. Okay. So now let us see the process of complete breakdown of the pyruvate. These 2 points are clear, or should I explain it again? Very important points. These 2 points are clear? Shravani? Okay, fine, good. Good. Tanuj Kumar, thank you. Fine. Now let us see. So the pyruvate, remember the site of glycolysis was cytoplasm. So it means the pyruvic acid is produced in the cytoplasm. For aerobic respiration, the pyruvic acid is first transported to the mitochondrial matrix and further process takes place in the mitochondria. So first of all, pyruvic acid is transported to the mitochondrial matrix. Then in the mitochondrial matrix, there is oxidative decarboxylation of pyruvic acid. This is sometimes called as the link reaction or gateway step. Okay. So pyruvic acid converted into acetyl-CoA. This process is called oxidative decarboxylation of pyruvic acid, and this process is commonly called as link reaction or gateway step. Why? Because after this another big process, Krebs cycle. So before this process, a big process, glycolysis. After this process, a big process, Krebs cycle, and it is linking the glycolysis to Krebs cycle. That is why this reaction is called as link reaction, this process. So see here in link reaction, what happens? Pyruvic acid plus coenzyme A plus NAD plus enzyme is pyruvate dehydrogenase, acetyl-CoA, which is a 2-carbon compound. Remember, from one pyruvic acid we have to remove 3 CO2. One pyruvic acid is 3-carbon compound. If 3 CO2 released means pyruvic acid completely broken down. So 1 CO2 is released here. So this is a 3-carbon compound. 1 CO2 released, so we get a 2-carbon compound acetyl-CoA, and we get NADH. Why we are calling this process as oxidative decarboxylation? Decarboxylation we can understand because there is release of CO2, that is decarboxylation. Why oxidation? Remember children, removal of hydrogen from a compound is the oxidation of that compound. If a compound is losing hydrogen to another compound, so the compound that is losing hydrogen is being oxidized, and one that is receiving the electron is being, hydrogen is being reduced. So see here, pyruvic acid is losing hydrogen to NAD. So pyruvic acid is being oxidized to acetyl-CoA while NAD is being reduced to NADH. So there is oxidation of pyruvic acid and there is reduction of NAD. So that is why we call this reaction as oxidative decarboxylation, because pyruvic acid is losing CO2, that is decarboxylation, and losing hydrogen, that is oxidation, and both taking place simultaneously. That is what is called as oxidative decarboxylation. Is this point clear? Fine. So this acetyl-CoA now goes to another process which is called as Krebs cycle. It is a cyclic process, okay? Krebs cycle, where further the remaining 2 CO2 of the acetyl-CoA will be removed. There will be complete breakdown of the respiratory substrate. So let us see. See, this is Krebs cycle. It is a cyclic process, and any cyclic process, children, there is always an acceptor. So acetyl-CoA is accepted by a 4-carbon compound, oxaloacetate, OAA. Acetyl-CoA is accepted by a 4-carbon compound, oxaloacetate, and in presence of water and enzyme citrate synthase, H2O is also used. Citric acid is formed, which is a 6-carbon compound. So remember, 4-carbon compound oxaloacetate, 2-carbon compound acetyl-CoA, and water is used, and we get a 6-carbon compound citric acid, and this reaction is called as condensation. This reaction is called as condensation. Now there is further conversion of the citric acid into alpha-ketoglutarate acid. So first of all, citric acid is converted into isocitric acid, okay? Citric acid is first, this is a type of isomerization. So citric acid is isomerized to isocitric acid. So this will also be a 6-carbon compound. Now oxidative isocitric acid again goes oxidative decarboxylation. See why? CO2 release decarboxylation, and hydrogen release taken by NADH to produce NADH, that is oxidation. So this is oxidative decarboxylation of isocitric acid to form alpha ketoglutaric acid. So one CO2 release, that is by alpha ketoglutaric acid, is a 5-carbon compound. Fine. Now, alpha ketoglutaric acid in the subsequent step is again undergoes oxidative decarboxylation. That is why there is only one 5-carbon compound in the Krebs cycle. So this again undergoes oxidative decarboxylation to produce a 4-carbon compound, succinyl-CoA. So see, by this time all the 3 CO2 from one pyruvate has been released. Remember, one CO2 was released in the link reaction, one CO2 released here and one CO2 released here. So complete breakdown of the pyruvic acid has taken place. Now this succinyl-CoA production is very, very important because in the next step, succinyl-CoA is converted to succinic acid. And children, why this is important? Because during this process, when succinyl-CoA is converted to succinic acid, there is SLP, substrate level phosphorylation. So first of all, in this process, first a GTP is formed. From GDP plus inorganic phosphate, a GTP is formed. And in a coupled step, this GTP is converted into GDP and the phosphate is taken by ADP to form ATP. So ultimately ATP is formed. So since ATP is formed, this step is again where I told you when from the substrate direct ATP is formed, it is called a substrate level phosphorylation. So this is a substrate level phosphorylation, fine. So ATP is formed here. Now succinate is converted into fumarate, fumarate is converted into malate, and malate regenerates the oxalo acetate. Okay, so malate regenerates the oxalo acetate. So what are we getting from one Krebs cycle? 2 molecules of CO2, 1, 2, 3, 3 molecules of NADH plus H plus, 1 molecule of FADH2 and 1 molecule of ATP. Okay, so this is the net gain from one acetyl-CoA. Remember, per glucose there will be 2 pyruvic acid, means 2 acetyl-CoA. It means per glucose there will be 2 Krebs cycle. So per glucose, if it is asked, you will have to multiply all this by 2. All this by 2. Because this is for one Krebs cycle, means 1 acetyl-CoA means 1 pyruvate. Per glucose 2 pyruvate means 2 acetyl-CoA multiplied by 2. So you will get for 1 glucose. So children, by this time, the complete oxidation of your respiratory substrate or glucose has taken place. All 6 CO2 have been released. But if you see the amount of ATP produced, we have got only 4 ATP per glucose till now. 2 ATP in glycolysis, 1 ATP, 1 Krebs cycle. Okay. But you know 1 glucose gives 38 ATP. So remaining 34 ATP, how do they come from? We will try to see this. But I'm taking a 5 minutes break. You also take a 5 minutes break. Have some snacks or something like that. It is around 10. We will be meeting at 10:05. I just will see. 10:05 again. Is it okay? 9:55 it is. By 10 we will meet again. 5 minutes break I'm taking. Any problem? Hello? Okay. So Okay, let's start. Are you back? Okay. Okay, fine. So children, remember, we say that complete breakdown of 1 glucose gives 38 ATP. But till now we have got only 4 ATP. 2 ATP glycolysis and 1 ATP per Krebs cycle per glucose 2 Krebs cycle, 2 ATP, 4 ATP. It means maximum ATP is somewhere still hidden that we have to get. This ATP is, children, hidden mainly in these compounds. See, this NADH and FADH2. If you see how many NADH we have at our disposal, starting from glycolysis, 2 NADH here, per glucose we are calculating, two NADH here, 4, and 3 2s are 6, 10 NADH and 2 FADH2. These are all high energy compounds. When this NADH and FADH2 will be oxidized, we will get the maximum ATP. And this takes place on the inner mitochondrial membrane. And this process is called as electron transport system and oxidative phosphorylation. Okay. And together it is called as terminal oxidation. So in aerobic respiration, exclusive steps of aerobic respiration are link reaction, second Krebs cycle, and third terminal oxidation, which involves two phenomena simultaneously, that is electron transport system and ATP synthesis oxidative phosphorylation. Remember, when in chloroplast due to light ATP was synthesized, we call it as photophosphorylation. Here we are getting ATP due to oxidation of compound, that is why we are calling it as oxidative phosphorylation. So see what happens. The site is the inner mitochondrial membrane, you can see here. So see, all the NADH and FADH2 that are produced in the earlier step, they are oxidized in the inner mitochondrial membrane. They are oxidized, so we get NAD plus 2 protons plus 2 electrons. Similarly, FADH2, FAD plus 2 protons plus 2 electrons. These electrons move on a chain of electron carriers on the inner mitochondrial membrane, creating the proton gradient, and the breakdown of proton gradient gives ATP according to the chemiosmotic hypothesis. Fine. Now the electron carriers on the inner mitochondrial membrane are in the form of 4 complexes and 2 mobile carriers. Let us see. This is complex 1, this purple one is complex 2, in between the green one is complex 3, and this pink one is complex 4. ETS only takes place at these 4 complexes, okay? 4 complexes. So all the NADH that was produced during the earlier step, it is oxidized on the complex 1, okay? That is why this complex is also called as NADH dehydrogenase complex, okay? The electron released is taken by ubiquinone. It is a mobile carrier present in the membrane, while protons are 4 protons are pumped into the intermembranal space. Remember, this is intermembranal space, and here will be the outer membrane. Fine. Now from ubiquinone, electrons are taken by complex 3, where it moves on chain of electron carriers and protons are pumped into the intermembranous space. Similarly, at complex 2, when succinate is converted to fumarate during Krebs cycle, there is production of FADH2. That FADH2 is oxidized at complex 2. Here also the electrons are taken by ubiquinone, okay, and they give to complex 3. It means complex 3 receives electrons from both complex 1 and complex 2, okay, and the protons are pumped into the intermembranous space, 4 protons. These electrons that complex 3 receives for complex 1 and complex 2, these electrons are taken by a mobile carrier cytochrome C, which moves on the outer surface of the inner mitochondrial membrane. This cytochrome C now moves and gives the electron to complex 4. Complex 4 has 2 copper centers and 2 cytochromes. CuA, CuB are the copper centers, and cytochrome are cytochrome A3. Okay, cytochrome A and cytochrome A3. So from cytochrome C, electrons are taken by complex 4, and from complex 4 ultimately the electrons are taken by oxygen. As you saw in chloroplast, the electrons were ultimately taken by NADP+. Here the electrons are taken by O2. So the ultimate electron acceptor is oxygen. That is why we call this phenomena as aerobic respiration. So although aerobic respiration started from Krebs cycle, but the role of oxygen comes now in the end. So when oxygen takes electrons from the ETS, it also pulls protons from the mitochondrial matrix and it forms water. This is called as metabolic water. And 2 protons are pumped into the intermembranous space. So see, same here it is also happening that due to electron transport, protons are being pumped from the matrix into the intermembranous space, creating a proton gradient. Similarly, here also in the inner mitochondrial membrane, there is a complex F0, F1 complex. F0 provides facilitated diffusion to protons, and F1, it has ATP synthase activity, it produces ATP. So due to breakdown of the proton gradient from the intermembranous space back into the matrix, energy becomes available. So the F1, which has ATP synthase, it combines inorganic phosphate with ADP to form ATP. For transport of 4 protons, if your NCRT, please correct this. New NCRT has given this. This should be 4 protons. And here also you have to correct 4 protons. So for the transport of 4 protons, 1 ATP is synthesized. Okay, 4 protons, 1 ATP is synthesized. Oxidation of 1 NADH will give 3 ATP, and oxidation of 1 FADH2 will give 2 ATP. So now we are in this marks the end of the aerobic respiration. Okay, so aerobic respiration, if we see common step glycolysis, then link reaction, then Krebs cycle, and then finally ETS and oxidative phosphorylation. Okay. Now we can calculate the respiratory balance sheet. Let us see. Anyone has any problem? Please let me know. Net ATP gain per glucose. Glycolysis by SLP to ATP. Link reaction by SLP 0 ATP. Krebs cycle by SLP to ATP. And electron transport system, this is very important. Electron transport system. First, glycolysis to NADH. Per NADH 3 ATP, so 6 ATP. Link reaction 2 NADH into 3 is equal to 6 ATP. Then Krebs cycle. Krebs cycle is also called as TCA cycle, tricarboxylic acid cycle, because the first product, citric acid, has a tricarboxylic acid, and it is also called as citric acid cycle. So Krebs cycle, because it was discovered by Hans Krebs. TCA cycle, because the first product is a tricarboxylic acid, that is why it is called as tricarboxylic acid cycle. Okay. Next, Krebs cycle. 6 NADH into 3 is equal to 18 ATP. 2 FADH2 into 2 is equal to 4 ATP. So how many ATP by SLP? Sorry, electron transport system, 34 ATP. Sir, after respiration, please take biomolecule cell cycle and cell division. Okay, fine. In growth, I will just explain 2-3 graphs and then we can move to, because I don't think there is requirement for PGR. So growth, I will just take 2-3 graphs and then we can move to biomolecules. No problem. Fine. Ajay. So by SLP, 4 ATP, and by ETS, 34 ATP. So this is 38 ATP per glucose. This question comes, in which step of respiration maximum ATP is produced? Our answer will be electron transport system, or you will call it oxidative phosphorylation. Fine. So with this, we have completed this process. Just 2-3 small things. But children, this calculation of 38 ATP is based on certain presumptions which are normally not met in the cell. That is why it is just a mathematical calculation. What are the presumptions? That there is sequential order pathway functioning, that first it should be glycolysis, then TCA cycle, and then ETS, but it does not happen. All processes take place parallelly. Second, the NADH synthesized in glucose, sometimes it does not enter the mitochondria. If it will not enter the mitochondria, it will not produce ATP, then you will not get 38 ATP. Third, none of the intermediate in pathway are utilized for synthesis. No, many intermediate compounds of the respiratory pathway are used for synthesis of other compound. For example, I told you that acetyl-CoA is used for synthesis of carotenoids and fatty acids. Similarly, alpha-ketoglutarate acid is used for the synthesis of amino acid glutamic acid. Oxaloacetic acid is used for the synthesis of other amino acids, fine, okay, aspartic acid and all. So that is why this is not possible. And fourth, only glucose is being respired. No, the respiratory substrate can be glucose, respiratory substrate can be fats, respiratory substrate can even be proteins in certain cases. So that is why this calculation is just based on certain presumptions. So these presumptions are not generally met in the natural system. That is why it is just a mathematical calculation. Fine. Amphibolic pathway, I have already told you. During respiration, the respiratory substrate is broken down to give energy. This is catabolism. See here. This is catabolic process of the respiratory pathway. Fats converted into fatty acid and glycerol. Glycerol will be converted into PGL, and PGL will enter the glycolysis. Fatty acid by beta oxidation will be converted into acetyl-CoA, and acetyl-CoA will enter the Krebs cycle. Carbohydrate, if it is starch or glycogen, first it will be broken down into glucose, and glucose will enter the glycolysis for further breakdown. Proteins, they are first broken down into amino acids, and amino acids can be used for the synthesis, can enter any pathway, okay? And amino acids can enter the any pathway. Mostly amino acids enter the Krebs cycle, but they can enter other pathway also. So this is all the catabolism. Anabolism, as I said, that this acetyl-CoA is used for the synthesis of fatty acids and carotenoids. Alpha-ketoglutarate acid is used for the synthesis of amino acid glutamic acid. Oxaloacetate is used for the synthesis of other amino acid like aspartic acid. Okay. Then it is also used for the synthesis of alkaloids and other compounds. So many intermediate compounds are used for the synthesis of other compound. That is why respiration is called as amphibolic pathway. Last thing we are coming is RQ, respiratory quotient. So during respiration, you see CO2 is evolved and O2 is consumed. Okay. So the volume of CO2 evolved upon volume of O2 consumed, this is called as the respiratory quotient. Now, if the respiratory substrate is rich in oxygen, then lesser oxygen will be consumed to release more CO2. Then the RQ will be higher, as in case of the glucose. Fine. So glucose. So RQ is 1. But if the respiratory substrate is poor in oxygen, then you will have to give more oxygen from outside to release CO2. Then the RQ will be less than 1. For example, fats and proteins. So fat have the RQ of 0.7, proteins have RQ of 0.9, and the carbohydrate or glucose has RQ of 1. So it depends upon the amount of oxygen present in the respiratory substrate. See here. To release 6 O2, 6 CO2, only 6 O2 is required. So 6 upon 6, 1. But you see here, in case of fat, to release 102 CO2, 145 O2 has to be given from outside. Why? Because your fat is poor in oxygen. That is why 102 upon 145, 0.7. And in between lies the RQ of the protein, which is higher than the fat but less than carbohydrate. So RQ of protein is 0.9. Fine. So with this, we have completed the chapter of respiration in plants. Just very basic 2-3 things of growth, and then we will move on to biomolecules. Plant growth and development. So growth is a permanent and irreversible increase in the plant organ or entire plant or a part of it or even a single cell. Permanent and irreversible increase. 3-4 points more. It is involved, it is associated with the synthesis of the protoplast. Third, it is a metabolic process that takes place at the expense of energy. And in living beings, growth is always from inside or intrinsic growth. Okay. Now, what are the special features of plant growth? In plant, growth continues throughout the life. This is called as the open form of growth. So plants show open form of growth. Meristems keep on adding new cells throughout the life of the plant. Second, in many plants, growth is in 2 phases. A primary growth, which is increase in length, and a secondary growth, which is increase in girth. No such primary and secondary growth is seen in case of animals. Fine. So this is how plant growth is different from the animal growth. In plant growth, there is increase in the number of organs as the plant grow, like leaves, flowers, fruits. In animal growth, number of organs remains constant, only the size increases. There is no increase in the number of organs. So these are the differences between plant and animal growth. Now coming to growth rate. Increase in growth per unit time is called as growth rate. Growth rate is of 2 types. First is called as the arithmetic growth. In case of arithmetic growth, it is constant. For example, if 1 hour, 1 meter height is increased in 2 hours, 2 meter height will increase in 3 hours, means growth per unit time is same. That is why it is called as it is called as the arithmetic growth. Okay. Why this arithmetic growth happens? Because out of the 2 daughter cells formed from a meristematic cell, only 1 remains meristematic, other it loses its meristematic activity. So per unit time, same number of new cells are added. That is why growth takes place at a constant rate. And if we mark a graph, it will give a linear curve. In case of arithmetic growth, we get a linear curve. And arithmetic growth can be represented by the formula LT is equal to L0 plus RT, where LT is the final length, L0 is the initial length, R is the growth rate, and T is the time interval during which you are measuring the growth. An example of such growth is growth of root, which shows constant growth in the soil. Fine. Second is geometric growth. In geometric growth, what happens? Both the daughter cells produced from a meristematic cell further divide. So with every per unit time, number of new cells added becomes double. So after first round, one new cell added. After second round, two new cells added. After third round, four new cells added. After fourth round, eight new cells added. So initially when the number of cells, dividing cells is less, growth rate is slow. This is called as the lag phase. But as the number of dividing cells increase, number of new cells formed exponentially increase. That is why this phase is called as the exponential phase or log phase. But due to limitation of the nutrients, because food is not unlimited. So due to limitation of the nutrients, this exponential phase cannot indefinitely continue. So a stationary phase comes. So we get a sigmoidal curve or S-shaped curve. See here. This graph is very frequently asked, so that is why I told you, very important. So initially growth rate is slow, lag phase, then suddenly growth very rapidly increases, exponential phase or log phase. But due to limitation of the food nutrients, the exponential phase cannot continue indefinitely. So a stationary phase comes. It shows the sigmoidal curve or S-shaped curve, which is characteristic of all organisms growing in the natural environment. See. An idealistic sigmoidal growth curve typical of cells in culture medium and many higher plants and plant organs, okay? They show sigmoidal growth curve. Now, if you see the formula of the exponential growth, only this much. If you see the formula of exponential phase, then W1 is equal to W0 e to the power RT. W1 is the final size. W0 is the initial size. E is the base of natural log. R is the relative growth rate, means above initial size, how much it has increased. That is, and T is the time interval. So this is called as the formula for the exponential growth. Fine. So relative growth rate and absolute growth rate, two things I will tell you, then we will finish. Suppose, see here, these are two leaves. This leaf, initially it was 5 centimeter and it became 10 centimeter. It means it increased by 5 centimeter square. So it increased by 5 centimeter square. It increased by 5 centimeter square. And this initial size was 50, final 55. So this also increased by 5 centimeter square. So this is called as absolute growth rate. Total growth over a period of time, irrespective of what was the initial size, this is called as absolute growth rate. So both leaf A and leaf B are showing same absolute growth rate, that is 5 centimeter square. But if you see, this has increased 5 upon initial size 5. Initial size was 5, upon 5 it has increased 5. So into 100. So 100%. But this has increased 5 centimeter upon initial size of 50 into 100. So what is the total? It is 10%. So this is called as relative growth rate. So relative growth rate is growth in unit time over initial size. Okay, total growth in unit time over initial size. So this has grown 100% over initial size. That is why absolute growth rate is 100%. This has grown only 10% of initial size. That is why the relative growth rate is 10%. So both leaf A and B have same absolute growth rate, that is 5 centimeters square. But leaf A, it has increased relatively more over initial size, so it's 100% relative growth rate, while leaf B has only 10% relative growth rate. Is it clear to everyone? Okay. Are you there? Hello. Sure. Okay, fine. So next we are coming to biomolecules. Carbon containing organic compounds that constitute the body of living beings are called biomolecules. These are also called as metabolites. These biomolecules can be classified on the basis of their size. Biomacromolecules 80 to 800 Dalton. Fine. These include amino acids, monosaccharides like glucose, then nucleotides and lipids. Although when we filter, if we crush a tissue and filter through a cheesecloth, the lipids they separate along with the biomacromolecules. Okay, but the size is less than 800 Dalton. That is why they are biomicromolecules, although they are separated with biomacromolecules. The reason is that phospholipids form the membrane, and when we disrupt the cell, these parts of the fragments of the membrane, they form vesicles, and these vesicles are retained on the cheesecloth. They do not get filtered down, but the actual size is less than 800 Dalton, so they come under although they are in the acid insoluble pool, but they are part of the biomicromolecules and not biomacromolecules. Biomacromolecules, more than 800 Dalton, or you can say more than 1000 Dalton, even more. So this includes proteins, polysaccharides, and nucleic acids. Proteins, polysaccharides, and nucleic acid, they come under the biomacromolecule. Now coming to biomicromolecules. First, amino acids. These are substituted methane. See here, if you see methane, if you substitute this with R group, substitute with this amino group, substitute this with acid group, you will get an amino acid. Okay, so amino acid, that is why they are called as substituted methane. And the carbon to which both amino and C group are attached, this is called as alpha carbon. So carbon on which both amino group and acid group are attached, or COH group itself, is called as alpha carbon. Now this R group can be as simple as hydrogen. As simple as hydrogen. Okay. For example, glycine. Or they can be methyl or hydroxymethyl. Let us try to see here. See. The R group is as simple as hydrogen, or this R group can be a methyl group, for example, alanine, or this R group can be hydroxymethyl, that is serine. Okay, fine. So this R group varies from amino acid to amino acid, and in some amino acid, instead of R functional group, an aromatic ring is present. These are called as the aromatic amino acids. Okay, aromatic amino acids. Can anyone tell example of aromatic amino acids? Tyrosine, tryptophan, and phenylalanine are the aromatic amino acids. Fine. Next is, if acidic group is more, acidic group is the COH group. If acidic group is more, then the such amino acids are called as acidic amino acids. For example, glutamic acid, aspartic acid, glutamic acid. These are acidic amino acids. If the amino group or basic group is more, then they are called as basic amino acids. You know why histone is a basic protein? Because it's rich in basic amino acid lysine and arginine. So lysine and arginine are basic amino acids. And if the number of amino group and acidic group are same, then such amino acids are called as neutral amino acids. For example, valine. So valine is a neutral amino acid. Glutamic acid and aspartic acid are acidic amino acids, and lysine and arginine are the basic amino acids. Fine. Next is that in amino acid, both amino group and carboxylic group can be ionized. See, if both are in the same amino acid, both are ionized, then this is called as zwitterionic form. So this is the zwitterionic form of the amino acid, where amino group is also ionized and carboxylic group is also ionized. Fine. So this is all regarding the amino acids. Next we come to nucleotides. Nucleotide consists of two parts, a phosphate group and a nucleoside. Nucleoside again consists of two parts. Pentose sugar and nitrogenous base. Okay, pentose sugar and nitrogenous base. So if you see, there are three components of a nucleotide: nitrogenous base, pentose sugar, and phosphate group. So let us see each one by one. Nitrogenous base, it has heterocyclic ring. It is of two types. First, purines. It has two rings or dicyclic. Nitrogen at position 1, 3, 7 and 9. So something like this. One, 3, 7 and 9. Okay. So nitrogen present at position 1, 3, 7 and 9, and it is a double ring structure. These are of two types, adenine and guanine. Both adenine and guanine are found in both DNA and RNA. Okay, so both purines are found in both DNA and RNA. Next is pyrimidines. It is single ring structure. Nitrogen at position 1 and 3. At position 1, 2, 3, 4, 5, 6. So nitrogen is at position 1 and 3. These are of three types. Cytosine, thymine and uracil. Cytosine is both in DNA and RNA. Thymine only in DNA and uracil only in RNA. Fine. Uracil only in RNA. Fine. So this is all regarding the nitrogenous bases. Next we come to pentose sugar. It is of two types. First, ribose sugar. So it has 5 carbons, 1, 2, 3, 4 and 5 carbon, that is why it is pentose sugar. And here at carbon 2, at C2, there is OH. So a pentose sugar which has OH at C2 is called as the ribose sugar. It is found in RNA. In RNA. And on the other hand, deoxyribose sugar. Here if you see one oxygen is missing. Here one oxygen is missing, that is why it is called as deoxyribose sugar, means one oxygen is missing at C2. So at C2 there is H instead of OH, and this is found in DNA. That is why DNA is called as deoxyribonucleic acid, and since RNA has ribose sugar, it is called as ribonucleic acid. So the main difference in DNA and RNA is due to the pyrimidine nitrogenous bases, because in DNA there is thymine and in RNA that is uracil. And second difference is due to the type of sugar. DNA has deoxyribose sugar while RNA has ribose sugar. Phosphate group. H3PO4. Okay. And due to this phosphate group, the overall charge on the DNA is negative. Okay, DNA or RNA, the overall charge is negative, and due to this phosphate group. So this is all regarding the different components. Now let us see a nucleoside. Suppose this is a pentose sugar. This is a nitrogenous base. This bond Between the pentose sugar and nitrogenous base, this is called as the N-glycosidic bond. Fine. Now remember, if the nitrogenous base is purine, then this bond is between C1 and N7. Okay. C1 of sugar and N7 of nitrogenous base. And if this nitrogenous base is pyrimidine, then it is a C1N1 bond. Are you understanding this point? Please let me know. Always the C1 of carbon will form a bond. If it is purine, then it will form with the 7th nitrogen of the nitrogenous base. So C1 and 7 bond. And if it is pyrimidine, then it will form with the N1 of the pyrimidine, then it will be C1N1 bond. This is a nucleoside. Nucleotide. So nucleoside plus phosphate group linked by phosphoester bond. Linked by phosphoester bond. So see here, this is N base, this is a nucleoside, and see here, fine. Now the phosphate group, OH, OH, OH and O. A water molecule is released from here. Okay. So the bond that is formed here, this PO bond, this bond is called as the phosphoester bond. So now entire structure is a nucleotide. So nucleoside plus phosphate group. Nucleoside plus phosphate group, this entire structure is called as nucleotide. Nucleotide. Fine. Now, how naming is done? N base, nucleoside, nucleotide. This question was asked last time. Okay, this question was asked recently. So if it is adenine, this will be called as adenosine. And this will be called as adenylic acid. If it is guanine, guanosine, guanylic acid. And so on and so forth. I'm not writing all of this. So this way you can name all the nucleosides and nucleotides, and this question is very frequently asked. So please see all these names. Fine. So this is all regarding the nucleotide. Then we come to lipids. Lipids. Water insoluble but soluble in organic solvents. Fine. That is why if your oil is there on your hand, you wash with water, it does not easily get removed. Fine. do not form polymers. Very important. Because you see, amino acids, they polymerize to form protein. Nucleotides, they polymerize to form DNA or RNA. Monosaccharides like glucose, etc., they polymerize to form polysaccharide. But there is no polymer of lipids. Lipids do not form polymers. Okay, remember this point. Fine. Do not form polymers. And types of lipid are fatty acid. Second, glycerol. Third, glycerides or fat. These are esters of fatty acid and glycerol. And fourth, fat-like substances. So they do not have fat, but their properties like fat. For example, cholesterol. Cholesterol comes under lipid, but it is not a fat, but it gives property like fat. So all these come under the lipids. So now let us start. See. Lipids are generally water insoluble. Water insoluble. They could be simple fatty acid first. A fatty acid has a carboxylic group attached to R group. So see, R group, okay, and COOH. So this is a fatty acid. This R group can be amino group, see, methyl group, ethyl group, or a long chain of CH. But this R group can have maximum 19 carbon. R group can be methyl group, ethyl group, or long chain of CH, but R group can have maximum 19 carbon. It means a fatty acid can have maximum 20 carbon because one carbon is of CH also, fine. Or higher number of CH2 group, 1 to 19 carbon. It means a fatty acid, the R group can have maximum up to 19 carbon. For example, palmitic acid has 16 carbon, including carboxyl carbon. It means the R group of palmitic acid has 15 carbon and 1 carbon of the COH, so 16 carbon. Similarly, arachidonic acid has 20 carbon. So that is the maximum one can have because R group will have 19 and 1 of COH, so 20 carbon. Fine, 20 carbon. So this is a fatty acid. Now, a fatty acid can be saturated or unsaturated. If a fatty acid has carbon-carbon double bond in it, then it is called as unsaturated fatty acid. And if there are no carbon-carbon double bond present in the fatty acid, then they are called as saturated fatty acid. So fatty acid could be saturated without double bond or unsaturated with one or more carbon double bond. If it is one carbon double bond, then it is called as MUFA, monounsaturated fatty acid. For example, oleic acid. And if it has many carbon-carbon double bond, then it is called as PUFA, polyunsaturated fatty acid. For example, arachidonic acid. And these fatty acids are good for our health. Unsaturated fatty acids are good for our health. Fine, okay. Another simple lipid is glycerol, which is a trihydroxypropane. So let us see. Let us see here. So this is first type of lipid, that is fatty acid. This is second type of lipid, that is glycerol. Okay, so trihydroxy alcohol. Fine. Okay. Third type of lipids are fatty acid plus glycerol. See here. Glycerides. Fatty acid plus glycerol. Linked by ester bond. So we can say that glycerides are esters of fatty acid and glycerol. So now they can be three types. And these are simple glycerides. Monoglyceride. One glycerol plus one fatty acid. Diglyceride. One glycerol plus two fatty acids. Triglyceride. One glycerol plus three fatty acids. Okay, so monoglyceride, diglyceride, and triglyceride. So let us see here. This is a picture of triglyceride. If you see here, this is one glycerol, this is one fatty acid, this is second fatty acid, this is third fatty acid. So one glycerol plus three fatty acid, it is a triglyceride. So this is a simple glyceride. Okay, a complex glyceride is where along with fatty acid and glycerol, some other compounds are also present. And a very good example is lecithin. See here. This is a complex glyceride because along with the fatty acid in glycerol, it also has nitrogenous compound and phosphorus containing compound phospholipid. So lecithin is a phospholipid because it is a lipid having a phosphate group. Along with that, it also has a nitrogenous compound which is called as choline. So lecithin has, if you see, one glycerol here, two fatty acids, one fatty acid and second fatty acid, one phosphate group, this is the phosphate group, and one nitrogen compound. So remember, there are one glycerol, two fatty acid, one phosphate group, and one nitrogenous compound choline. So this is the structure of the phospholipid lecithin, and you know the lipid bilayer of the cell membrane is lecithin. So lipid bilayer of the cell membrane is made up of lecithin. Fine. So it is a phospholipid. Next, fat-like substances which are not actually fat because they do not have fatty acids, but their property is similar to fat, which is cholesterol. So you see, cholesterol is a fat-like substance. Fine. So with this, we have completed the biomacromolecule, that is amino acids, nucleotides, and lipids. Is it clear? Can we proceed? Rishita, are you there? Okay, okay. Actually, your messages were not coming at all, so I thought there's some problem. Just wait for 2 minutes. We are continuing. So biomicromolecules we have completed. Okay. Now we are coming to biomacromolecules. In biomacromolecules we are starting with proteins. Proteins are polypeptide, means many amino acid linked to each other through peptide bond. This is called as polypeptide. So proteins are polymers of amino acids. And proteins are heteropolymers because they are made up of different types of amino acids. So all proteins are heteropolymers. Fine. Okay. They are linear chain of amino acid linked by peptide by they are shown in figure. Fine. Each protein is a polymer of amino acid. There are 20 types of amino acid which participate in protein synthesis. Some of these amino acids our body can synthesize. Some of them we are dependent on our dietary protein. They are required by our body, but we do not synthesize. So we are dependent on our dietary protein, and these are called as the essential amino acids. Okay, these are called as essential amino acids. A protein is a heteropolymer and not a homopolymer. Why? Because it is made up of different types of amino acid. A homopolymer has only one type of remaining chain. The information. See. Even that certain amino acids are essential for our health and they have to be supplied through our diet because our body is not able to synthesize them. Hence, dietary proteins are the source of essential amino acids. Therefore, amino acids can be essential or non-essential. So, if I give you a list of essential amino acids, phenyl, alanine, methionine, threonine, histidine, isoleucine, leucine, lysine, valine, okay. Now these are the essential, but this arginine, this is essential only in the infants. As we grow adult, we are able to synthesize them. So these are the list of the essential amino acids. Fine. Next, proteins have multiple functions in our body. They can be enzymatic function. They can function as the immunoglobulins, that is the antibodies. They can have receptor function in our body. They can be hormones. So this is a list given that collagen, intercellular ground tissue, trypsin is an enzyme, insulin is hormone, antibodies, immunoglobulins, receptor, sensory receptor that sense the hormones. This GLUT4 is very frequently asked. GLUT4 is a glucose transporter present in the membrane through which glucose comes inside the body. So this GLUT4 is a membrane transport proteins which facilitates transport of glucose in our body. Fine. Okay. So this is important. Now, the most abundant protein on earth is the enzyme Rubisco, about which we studied in the Calvin cycle, the first enzyme of Calvin cycle, Rubisco. It is the most abundant protein on earth. Why? Because it is a slow enzyme. So because the enzyme is slow, so to increase the rate of photosynthesis, the amount of enzyme is increased. That is why it is the most abundant. In animal world, the most abundant protein is the collagen. Fine. Okay. So see here. Collagen is the most abundant protein in animal world, and Rubisco is the most abundant protein in the whole of the biosphere. Fine. Okay. Now, similar light here, we also let us do the structure of protein. protein structure. First, primary structure. It is a linear chain of amino acids. It is a linear chain of amino acids. Left hand side is N-terminal because free NH2 is present, and at right hand side C-terminal because free COH is present. So left hand side of this is called as a polypeptide. This is amino acid and this is the peptide bond. This is the primary structure of protein. It has an N-terminal and a C-terminal. Left hand side is N-terminal and right hand side is T-terminal. Second, secondary structure. In secondary structure is of three types. First, alpha helix. And stabilized by hydrogen bonds. Okay. And remember, in protein, there is only right-handed helix. In protein, left-handed helix is not found. Fine. Only right-handed helix, alpha helix. Second, beta plate. It is folded like a plate upon itself. Stabilized by H bonds. So these are the two structures, alpha helix and beta plate. They were given by Linus Pauling. Then our Indian origin scientist, Dr. G. N. Ramachandran, he discovered the triple helical structure of collagen. Okay, so triple helical structure of collagen. Triple helical structure of collagen. So three polypeptide chains are helically coiled among each other. So this is the triple helical structure of collagen, which was discovered by Dr. G. N. Ramachandran. Fine. So these are all secondary structure of proteins. Next we come to tertiary structure. Polypeptide is folded upon itself like a ball of wool. You see the wool ball which is used for knitting purpose. Okay, so it is something like this. N-terminal, C-terminal. Fine. And this tertiary structure is very essential for many functions of protein which primary structure and secondary structure cannot do. For example, enzymatic function. Okay, because for a protein to function as enzyme, it should have these pockets or crevices which are called as active site. So these active sites will not be formed in primary or secondary structure. So that is why tertiary structure of protein is very important for certain functions of protein. And as I said, one of them is the enzymatic function of protein. Fine. So this is the tertiary structure. And fourth, quaternary structure, when more than wool balls are connected to each other. For example, like this. They can be in this form or they can be in the linear form, but more than wool balls when like structure, but connected to each other, they form the quaternary structure. And very good example is hemoglobin. It has two alpha chains and two beta chains. In adults, remember, okay, in infants it is gamma chain, and in adults, human hemoglobin, there is two alpha chains and two beta chains, but in infants it is gamma chains. Fine. So let us see here. So this is all regarding the protein. Next, carbohydrates. These are a class of biomolecules. Okay, or you can say polysaccharides. polysaccharides. Class of biomolecules called as carbohydrates. These are polymers of monosaccharides like glucose, fructose, ribose, etc. Fine. So let us see this monosaccharides. See, this is a glucose and this is ribose, because OH is present, this is ribose. And when they will polymerize by glycosidic bond, they will form the polymers or the polysaccharides. So let us see the different types of polysaccharides. And in a polysaccharide, suppose this is a polysaccharide, the left hand side is called as non-reducing end and right hand side is called as reducing end. Remember why it is so? Because the C1 of the sugar should have free OH. If the C1 of the sugar has free OH, then that end will be the reducing end. And if the CN of the sugar is involved in glycosidic bond formation, then it will be non-reducing end. So right hand side, C1 of the sugar is free, that is why it is the reducing end. And left hand side, the C1 of the sugar is involved in glycosidic bond formation, that is why it is a non-reducing end. Fine. So let us see some of the polysaccharides. See, first of all, cellulose. Cellulose is a homopolymer. Why it is homopolymer? Because it is made up of only beta glucose. Only one type of monosaccharide, that is why it is called as a homopolymer of beta glucose. Okay. A starch is variant of this. Why variant of this? Because starch is a homopolymer of alpha glucose. So that is why we are calling starch as a variant of cellulose, because cellulose is a homopolymer of beta glucose and starch is a homopolymer of alpha. Another homopolymer of alpha glucose is glycogen. So both glycogen and starch are homopolymers of alpha glucose, while cellulose is a homopolymer of beta glucose. And cellulose is called as a structural polysaccharide because it forms the cell wall, structure of the cell, while starch and glycogen are called as storage polysaccharide because plants store food in the form of starch while animals store food in the form of glycogen. That is why glycogen and starch are examples of storage polysaccharide, while cellulose is the example of structural polysaccharide. Now, for a polysaccharide to retain iodine, it should have a complex helical structure. Cellulose does not have a complex. Very, very common question it is asked. Okay. Starch forms helical secondary structure. In fact, the starch can hold iodine molecule in the helical protein. The starch I2 is blue in color. Cellulose does not contain complex helical structure, hence cannot hold iodine. So that is why if you see add iodine and blue color comes, we say that there is confirmation of presence of starch. But for the hold iodine, it should have a complex helical structure. Cellulose does not have complex helical structure, that is why it cannot store starch. So all three, that is cellulose, starch and glycogen, are polymers of glucose. While another one, inulin, it is a polymer of fructose. Inulin is a polymer of fructose. And inulin is also stored in the form of food in roots of plants like dahlia. So inulin is also a storage polysaccharide. So remember, cellulose is a structural polysaccharide, while starch, glycogen, and inulin are storage polysaccharide because food is stored in that form. Okay, fine. Now these, up till now we were studying about the simple polysaccharides. So cellulose, starch, glycogen, inulin. They are only made up of monosaccharides, means unmodified sugar. So they are called as simple polysaccharides. And if a polysaccharide is made up of modified sugar, then it is called as complex polysaccharide. For example, see, there are more complex polysaccharides in nature. They have a building block, amino sugars and chemically modified sugars. For example, see, modified sugars, glucosamine, N-acetylglucosamine. If the polysaccharide is made up of these sugar, then it is called as complex polysaccharide. For example, chitin. It is made up of N-acetylglucosamine. It is a homopolymer of N-acetylglucosamine. So that is why, since it is made up of chemically modified sugar, it is called as the complex polysaccharide. And it is also a structural polysaccharide because it forms the cell wall of the fungi. Fine. So with this, we have completed the polysaccharides. Any question you want to ask in polysaccharides, or we can move forward? Then last we are going to do is nucleic acid. Nucleic acid. Heteropolymer of deoxyribonucleotides. Okay, heteropolymer, such nucleotide which is containing deoxyribose sugar, deoxyribonucleotides. So if we see here, it is a double helical structure having backbone of phosphate and sugar. The diameter of the helix is 20 angstrom or 2 nm. One complete turn or one pitch is 34 angstrom or 3.4 nm. And in one complete pitch, there are 10 base pairs. And the distance between adjacent base pairs is 3.4 angstrom or 0.34 nanometer. Fine. So this is the dimension of the double helical structure of DNA. The diameter you know, the length of one complete turn you know, number of base pairs in one turn. So in 3.4 nm there are 10 base pairs, so distance between adjacent base pairs is 0.34 nm. And it is a backbone of phosphate and sugar. Let us see. Phosphate, sugar, phosphate, sugar, phosphate, sugar. Okay. Nitrogenous base are attached to the sugar. So this end is called as a 3' end and this end is called as 5' end. So here it is anti-parallel, this will be the 3' end and this will be the 5' end. So we say that the backbone is made up of sugar and phosphate and the nitrogenous base are projected at right angle towards inwards. Okay. The two chains are held together by hydrogen bond. Always adenine pairs with thymine with 2H bonds and guanine pairs with cytosine with 3H bond. This is called as complementary base pairing. Okay. So always a purine pairs with pyrimidine, that is why the diameter is constant. This is called as complementary base pairing. So this is all regarding the structure of DNA. Now if you see the angular rise per base pair, so one complete turn is 360 degree and in 360 degree there are 10 base pairs, so angular rise per base pair is 36 degree. So in 360 degree there are 10 base pairs, so angular rise per base pair is 36 degree. Let us see this all this here. See, nucleic acids exhibit a wide variety of secondary structure. For example, one of the secondary structure exhibited by DNA is the famous Watson-Crick model. This model was given by Watson-Crick. This model says that DNA exists as a double helix. The two strands of polynucleotide are anti-parallel. Why? Because orientation is opposite. This diagram I have drawn you, just have drawn this diagram, okay? Are anti-parallel. Run in opposite direction. The backbone is formed by sugar phosphate sugar chain. The nitrogenous bases are projected more or less perpendicular to this backbone but face inside. Adenine and guanine of one strand compulsory base pair with thymine and cytosine respectively on the other strand. Fine. There are 2 hydrogen bonds between A and T and 3 hydrogen bonds between G and C. Each strand appears like a helical structure. Each step of ascent is represented by a pair of base at each step. See, at each stage of ascent means moving up. At each step of ascent, the strand turns 36 degree. Why? Because there are 10 base pairs, total 360 degrees. So per base pair, the turn is 36 degree. One full turn of helical strand would involve 10 steps or 10 base pairs. Attempt drawing a line diagram. The pitch would be, pitch means one complete turn is called as pitch, 34 angstrom. The rise per base pair is 3.4 angstrom. This form of DNA with the above mentioned salient features is called as bDNA. Okay. In higher classes, okay, fine, bDNA. And it is right-handedly coiled DNA. So with this, we have completed biomacromolecules. Now, another thing which is important in this is enzymes. If you can pinpoint me in enzyme, what should I discuss? If you can pinpoint me specifically in enzyme, what part should we take? Complete enzyme, it will take a long time. Anyway. So children, enzymes are the catalyst of the biological system. Okay. In biological system, chemicals that increase the rate of a reaction without getting involved in the reaction, they are called as the enzymes. So they are the catalyst of the biological system. Chemically, enzymes, almost all enzymes are proteins, but there are some exceptions also. In certain cases, the nucleic acid RNA functions as enzyme. So if RNA functions as enzyme or RNA shows a catalytic role, they are called as ribozyme. For example, peptidyl transferase. Okay, fine. Okay. Factors affecting enzyme activity and classification. Okay, fine. Just basic information and then I'll do. Okay. There are some nucleic acid that behave like enzymes. As I said, ribozyme. One can see an enzyme like any protein has a primary structure. Amino acid sequence of the protein enzyme like any protein has the secondary structure and tertiary structure. When you look at tertiary structure, you will notice that backbone of the protein chain folds upon itself, the chain crisscross itself and hence make rise. That is why I said the tertiary structure of the protein is essential for the enzymatic activity. In primary and secondary structure, you will not see the binds. Okay. Now, generally, since enzymes are proteins, so they show their activity at moderate temperature. They show their activity at moderate temperature. If temperature becomes more than 40 degrees centigrade, the enzymes undergo denaturation. The enzymes undergo denaturation. But there are certain enzymes that are thermostable enzymes. Remember one used in the PCR, Taq polymerase. Okay. So these enzymes are called thermostable. They can remain active even between 80 to 90 degrees centigrade. An example, as I said, up to 80 to 90 degrees centigrade. An example is Taq polymerase, which is obtained from the hot vent and sulfur springs. Fine. Okay. Now, three things have come to my mind. Factors affecting enzyme activity, classification, and cofactors. Okay, fine. So let us see. I will explain this graph also because many people have them. That how enzymes increase the rate of reaction. Let us see how enzymes increase the rate of reaction. Remember, for any chemical reaction, from substrate to product directly does not take place. From substrate, it will first reach the many intermediate stages, which is called as transition state, and then to product. So substrate to transition state to product. Whether it is endothermic reaction or exothermic reaction, in both type of reaction, a very large amount of energy is required to reach the transition state. And this energy is called as activation energy. So due to this very high demand of energy for the substrate to reach the transition state, very few substrate molecule get converted and reach the transition state. That is why very few substrate molecule get converted into product. That is why the rate of a biochemical reaction is very, very slow. But if enzyme is present, enzyme lowers the requirement of activation energy. So to reach the same transition state, now very less energy is required in the presence of enzyme. So now if less energy is required, more substrate gets converted into the product because less energy is required to reach the transition state. So if you are asked how enzymes increase the rate of a biochemical reaction, enzymes increase the rate of a biochemical reaction by lowering the activation energy. So to reach the transition state, now very less energy is required. So that is why large amount of substrate, they get converted into transition state and ultimately get converted into the product. So final line is enzymes increase the rate of biochemical reaction by lowering the activation energy. Fine. Now factors affecting enzyme activity, let us see. So see here, if we see temperature, factors affecting enzyme activity, temperature and velocity of the reaction, that is activity. If temperature is very low, then enzyme will remain inactive. So if you increase the temperature, enzymatic activity will increase, okay? Increase and keep on increasing. But a time will come when further increase in temperature will cause denaturation of the enzyme. As a result, the enzyme activity will decrease, okay? So this temperature is called as the optimum temperature. It means this is the temperature which is best suited for the enzyme activity. Okay. So below this, enzyme activity will be low due to inactivation, and beyond this, enzyme activity will be low due to denaturation. So if we make a graph for temperature, we get a bell-shaped curve, and same we get for pH also. Because every enzyme shows its best activity in a narrow range of pH. So if you decrease the pH below this, then also enzyme activity will decrease. And if you increase the pH beyond this, then also enzyme activity will decrease. So both temperature and pH give bell-shaped curve. For example, you know that the lysosomal enzymes, they are called as acid hydrolases. So they show their best activity between 4 to 5 pH. Now, if you will lower the pH further down, there will be decrease, and if you increase the pH to alkaline side, then also there will be decrease. So both temperature and pH, they show bell-shaped curve. Now, substrate concentration. Suppose this is an enzyme molecule which has so many active sites. This enzyme has so many active sites. You will add substrate. Some substrates will bind to active site. Enzymatic activity will increase. You give some more substrate, more active sites will be occupied, further increase in the enzymatic activity. You give some more substrate, more active sites will occupied, further increase in the enzymatic activity. You give more substrate, a time will come when all active sites are now occupied. Now, if you further increase the substrate, there is no active site free to use this substrate. It means now there will be no further increase in the rate of the enzymatic activity. Why? Because now you will say the enzyme has got saturated with substrate. Fine. So that is why if you make a graph for substrate concentration and velocity, so with the increase in substrate concentration, the velocity of the reaction will increase because more and more substrate will bind to more active sites increase. But a time will come when the enzyme, all the active sites are now occupied by substrate. Enzyme is saturated with substrate. So with further increase in substrate concentration, there will be no further increase in the rate of reaction. Fine. So you will get this graph. Now it means now enzyme is working at its maximum velocity. This is called as Vmax. Fine. And half of this Vmax, this is called as Vmax by 2. Now, if you see, this much substrate was required for the enzyme to reach Vmax by 2. This is called as Km, Michaelis-Menten constant. So Km, what is Km? So Km is that substrate concentration at which enzyme is working at half of its maximum velocity, or Vmax by 2 is called as Km. Now, why we calculate Km? It means if enzyme is using lesser substrate to reach Vmax by 2, it means enzyme has a stronger affinity for the substrate. Okay. If enzyme uses large amount of substrate to reach Vmax by 2, it means enzyme has a lesser affinity for the substrate. So Km is an indicator of the affinity of the enzyme for substrate, and it is inverse. Lower the Km, more is the affinity of enzyme for substrate. Higher the Km, lesser is the affinity of the enzyme for the substrate. This point is clear to everyone. What is Km and why it is calculated? Next are inhibitors. These are chemicals that bind to the enzyme and retard its catalytic activity. These are chemicals that bind to the enzyme and retard its catalytic activity. Now, this inhibition is of two types. First is competitive inhibition. inhibitor is structurally similar to substrate. So both substrate and inhibitor compete to bind to active site of enzyme. Because both are structurally similar, so there is a competition between the inhibitor and the substrate who will bind to the active site of enzyme. But the problem comes here, children. See here. Suppose this is enzyme. And this is substrate. If substrate binds to the enzyme, enzyme substrate complex. Okay. This will result in product plus enzyme. But if inhibitor is structurally similar, so inhibitor can also bind to the enzyme. This will lead to enzyme inhibitor complex. No product form. Since product will not be formed in second case, that is why what will happen? The activity of the enzyme will decrease. More the inhibitor, more will be active site will be wasted by the inhibitor, so lesser will be the rate of the reaction. Fine. So both inhibitor and the substrate bind to the same common site of the enzyme because inhibitor is structurally similar to the substrate. But when the substrate binds to the enzyme, product is formed. When inhibitor binds to the enzyme, product is not formed. As a result, rate of reaction is decreased. So it means, so can this process be reversed? Yes. Even in the presence of inhibitor, enzyme can reach Vmax by 2, but by using more substrate. Why? Because if there is competition between the inhibitor and substrate, you increase the substrate concentration, then substrate will win this competition, but it will be requiring more substrate to reach the Vmax by 2. See here. This is Km without inhibitor. Now, if you add inhibitor, then more substrate will have to be added for the enzyme to reach the Vmax by 2. So, if enzyme is more substrate is required, then what happens? Km increases with inhibitor. So, in case of competitive inhibition, the Km of the enzyme increases because now more substrate has to be given for the enzyme to reach Vmax by 2. Now, example of competitive inhibition is malonate is competitive inhibitor of enzyme succinate dehydrogenase. If you remember, there is an enzyme in the Krebs cycle which is called a succinate dehydrogenase. If you give malonate competes with succinate to bind to the active site of enzyme. As a result, the catalytic activity of the succinate dehydrogenase is decreased. Second, such competitive inhibition is also used to check bacterial infection. So this is all regarding the competitive inhibition. Next is non-competitive irreversible inhibition. non-competitive reversible inhibition. The inhibitor binds at a site other than active site on enzyme. So it does not compete with substrate, but binding of inhibitor permanently inactivates the enzyme. That is why we are calling it as irreversible. Okay, so let us see. Suppose this is active site. This is substrate. This is inhibitor. If inhibitor binds to this site, it permanently changes the conformation of the active site. So substrate cannot bind to the active site. Okay, so see, inhibitor is binding at a different place, not at the active site. That is why there is non-competitive. But binding of inhibitor to the enzyme is permanently inactivating the active site, because the substrate cannot bind, so rate of the reaction will decrease. So in case of this inhibition, what happens? The enzyme fails to reach the Vmax. So this is without inhibitor and this is with inhibitor. Since enzyme nature is permanently destroyed, so enzyme fails to reach the Vmax. That is why in case of non-competitive inhibition, the Vmax is decreased. Now where it is found? So if you see here, I will bring this picture again. You all know that if we consume cyanide, we will die. But how consuming cyanide brings us death, let us try to see. And this is example of non-competitive irreversible inhibition. You know, during ETS, the electrons are taken by complex 4 and ultimately by oxygen. Now, if you give cyanide, cyanide binds here. Cyanide binds here and prevents the transportation of electron to oxygen. It means due to binding of cyanide, the ETS will stop. If ETS will stop, then proton gradient formation will stop. And if proton gradient formation will stop, then ATP synthesis will stop. And if ATP synthesis stops, body will not be able to perform work and we will die. Because for every metabolic reaction, we require energy source. So cyanide, when binds to the complex 4, it prevents the transportation of electrons from the complex 4 to the oxygen, thus stopping the ETS. Due to stoppage of ETS, there is stoppage of ATP production, and due to stoppage of ATP production, it ultimately leads to death. So this is a non-competitive irreversible inhibition, cyanide binding to the complex 4 of the ATS of mitochondria. Is it clear? I'm not getting any response from your end, so I'm not able to understand. Is there any problem in communication? So this is all regarding factors affecting enzymatic activity. Now coming to classification. See, There is an organization called as Enzyme Commission. This Enzyme Commission has given a 4-digit number to every enzymes. For example, a coded disease, 4-digit number to every enzymes. What they have done, on the basis of the type of reaction the enzymes perform, they have classified all enzymes into 6 classes. Okay, class 1, class 2, class 3, class 4, class 5, 6. Okay. And this each class has been further divided into 4 to 13 subclasses. And on this basis, each enzyme has been given a 4-digit number. For example, suppose a number is an enzyme commission number of an enzyme is 3251. So we are just concerned with the first letter. Okay. So first letter represents the class of enzyme. Enzyme belong to which class? So this first digit can vary from 1 to 6 because all enzymes have been divided into 6 classes. So I will give you a shortcut if you can remember it. Just remember a word orthyl. This is class 1 called as oxidoreductase. This is class 2 called as transferase. This is class 3 called as hydrolase. This is class 4 called as lyase. This is class 5 called as isomerase. And this is class 6 called as ligase. So these are the 6 classes. So all enzymes on the basis of their function have been divided into 6 classes. Okay, and each class has been further divided into 4 to 13 subclasses. And on this basis, Enzyme Commission has given a 4-digit number to each enzyme. The first digit represents the class of the enzyme. For example, it means if the enzyme, as I said, 3241 it is written, it means it is a hydrolase. It is a hydrolase. If some enzyme is given 5123, it means it is a isomerase enzyme. So this way you can understand. Fine. Now let us try to see the 6 classes. So first class is oxidoreductase. Oxidoreductase means one will be oxidized and another will be reduced. I told you release of hydrogen from a compound is oxidation and acceptance of hydrogen from a compound is reduction. So suppose this has hydrogen. So if it is giving this hydrogen to this one, so this one will be reduced and this one will be oxidized. So one that received, so S reduced, X oxidized. Okay, it gave it hydrogen to this. So this reduced one became oxidized and this oxidized one became reduced. Fine. For example, as I said, that suppose pyruvic acid NAD plus, it gives it hydrogen to NAD plus to get reduced into NADH and it converted into acetyl CoA. So there is oxidation of pyruvic acid because it has lost hydrogen and there is reduction of NAD plus because it has accepted hydrogen. So transfer of hydrogen, okay. That is why it is also called as dehydrogenase, means loss of hydrogen. Enzyme which catalyze oxidation reduction between two substrate. Okay, this is called as oxidation. So all dehydrogenase enzyme, alcohol dehydrogenase, pyruvate dehydrogenase, lactic acid dehydrogenase, they all will come under the first, that is oxidoreductase or dehydrogenase. Fine. Next, transferase. If one compound is transferring group to another compound, but not hydrogen, because transfer of hydrogen, we have already said, transfer of hydrogen is oxidation reduction. But apart from hydrogen, if any other group is transferred from one compound to other compound, then the enzyme catalyzing is called as transferase. For example, see, this has G. This G was transferred to S star. So S took the G, so this S star became, okay, so this is transfer. For example, I will tell you, glutamic acid plus oxaloacetic acid. Glutamic acid is amino acid, oxaloacetic acid is an amino acid. When glutamic acid transfers its amino group to oxaloacetic acid, it forms another amino acid, aspartic acid. So amino group was transferred from glutamic acid to oxaloacetic acid. As a result, oxaloacetic acid converted into another amino acid, aspartic acid, and the enzyme involved is called as the transferase. Okay, amino transferase. Okay, amino transferase. I mean, because it is transfer of amino group, that is why it is called as amino transferase. Amino transferase. So transfer of any group other than hydrogen from one compound to other compound, the enzyme that is catalyzing is called as transferase. Hydrolase. If by using water an enzyme is causing breakdown of a bond, this is called as hydrolase. Remember children, all complex biomolecules, if protein is being broken into amino acids, water is used, hydrolysis. If a polysaccharide is being broken into monosaccharide, this is hydrolase. Okay, so that is why all digestive enzymes come under hydrolases. Okay, all digestive enzymes come under hydrolases. For example, suppose starch, amylase plus H2O N, and this will result in glucose N amount. So by giving water, we are breaking the glycosidic bond in starch and converting into glucose, enzyme amylase. So amylase is a hydrolase. So when water is used to break bonds of a polysaccharide to convert them monosaccharide, it is called as hydrolases. So all digestive enzymes come under hydrolases. Lyases are enzymes which do not use hydrolysis. Okay, enzyme that catalyze removal of group from substrate by mechanism other than hydrolysis, and it always results in carbon-carbon double bond. See, so hydrolysis is not done, group is removed, see XY group removed, and it results in carbon-carbon double bond, then it is called as hydrolysis. For example, there is an enzyme called as enolase. Then fourth group is isomerase. Isomerase, you know, glucose converted into fructose, this is isomerization. So isomerization can be optical, it can be geometric. Okay, fine. Enzyme catalyzed into conversion of optical, geometric or position isomerization. Fine, position. See here. A very good example you see. 3-PGA. Enolase converted into 2-PGA. So this is a position isomerism because just the phosphate group was transferred from third carbon to second carbon. So this is a position isomerism. Similarly, see, phosphoglyceraldehyde, triose phosphate isomerase converted into dihydroxyacetone phosphate. This is an aldehyde, this is a ketone. Aldehyde being converted into ketone with the help of an enzyme. This is again isomerization. So all enzymes that catalyze such reaction are called as isomerases. Okay, fine. Last is ligases. Ligation means to join. Remember, ligation means to join. So if enzyme is catalyzing joining of two compounds by forming either CO bond, CS bond, CN bond or PO bond, then enzyme is lysis. For example, DNA polymerase, okay, or you can say the enzyme DNA ligase, very good enzyme. One DNA molecule is joined to another DNA molecule by ligases, you know, DNA ligase. So these are very good example of ligases. So these are the 6 classes of the enzyme. Now coming to cofactors. Cofactors I will tell you. See, enzyme, most of enzyme have 2 parts. A protein part or protein moiety called as apoenzyme and a non-protein part which is called as cofactor. So apoenzyme plus cofactor are together called as holoenzyme. What is the meaning of holo? Holo means complete. So protein part is apoenzyme, non-protein part is cofactor, and complete enzyme is whole enzyme. If this non-protein part or cofactor is removed, the enzyme loses its catalytic activity. The enzyme loses its catalytic. That is why this cofactor is very, very important. Now, types of this cofactor, see. Cofactor can be of three types. First, prosthetic group. If it is organic compound and tightly bound to the apoenzyme, it never separates from the apoenzyme. It is an organic compound tightly bound to the apoenzyme, then such cofactor is called as prosthetic group. For example, heme in the enzyme catalase and peroxidase. This heme is a part of the active site of enzyme. So if you will remove the heme, the active site of the enzyme will be disturbed and substrate will not bind to it. So organic compound firmly or tightly bound to the apoenzyme is called as the prosthetic group. Heme is a very good example in both catalase and peroxidase. Second, coenzyme. It is also organic compound but transiently bound. Transiently means when enzyme catalysis has to take place, it will bind and then it will separate. So transiently bound opposite to permanently bound. So at the time of enzyme catalysis, it will bind to the apoenzyme and then it will separate. So if the cofactor is transiently bound to the apoenzyme, then it is called as the coenzyme. It is called as coenzyme. Second thing about this is that same cofactor can function as coenzyme for many reactions. For example, very good example is NAD. NAD is the cofactor for the enzyme pyruvate dehydrogenase also, alcohol dehydrogenase also, alpha ketoglutaric acid dehydrogenase also, and malate dehydrogenase also. All use NAD as the cofactor. So same coenzyme can function as cofactor for many enzymes. Third, most of them have vitamins as a constituent. For example, in NAD, niacin is present. Okay, so this is third point. So 3-4 points. Transiently bound, organic compounds first. Second, transiently bound to the apoenzyme only during enzyme catalysis. Third, same cofactor can function as coenzyme for many enzymes. And fourth, most of them contain vitamins. Okay. And third is metal ions. In many cases, metal ions also function as the cofactor. So these metal ions, they form a coordinate bond with the active site of enzyme, and also they form coordinate bond with the substrate. See here. A number of enzymes require metal ions for their activity, which form coordinate bond with the side chain of the active site and at the same time form one or more coordinate bond with the substrate. Example is zinc is a cofactor for the proteolytic enzyme carboxypeptidase. This question is important, it can be asked. Okay, fine. So remember, zinc is a cofactor for the digestive enzyme carboxypeptidase. Many such examples are there. For example, magnesium is a cofactor for both enzymes of dark reaction, that is Rubisco also and peptase also. Fine. So many such examples are there. Molybdenum is a cofactor for nitrogenase enzyme. Nitrogenase enzyme, molybdenum. So many such examples are there. So this is all regarding children cofactors and also biomolecules chapter. Anything else you want in biomolecules, please tell me. Otherwise, I will move to another chapter. Okay. Cell cycle and cell division. So remember, cell cycle is a series of genetically controlled steps. Cell cycle is a series of genetically controlled steps where a newly formed cells enlarges its size, duplicates its genomic content. Genomic content means the chromosomes present in the nucleus, duplicates its cytoplasmic content, that is the organelles, and then further divides to form two daughter cells. Okay. So again, its cell cycle is a series of genetically controlled steps where a newly formed cell enlarges in size, duplicates its genomic content, duplicates its cytoplasmic content, that is organelles, and further divides to form two daughter cells. Fine. Okay. And it is a orderly phenomena. Fine. A distinct cell cycle is present only in eukaryotes. Okay. A distinct cell cycle is present only in eukaryotes. Okay. Now, in eukaryote, there are mainly two types of cell divisions. Mitosis and meiosis. Now, coming to mitosis, it is a type of cell division where a mother cell divides to form two genetically identical daughter cells. Chromosome number in both daughter cells is same as in mother cell. Hence, it is also called as equational division. Hence, it is also called as equational division. Fine. Now, cell cycle has two distinct phases. First is called as the interphase, and second is called as M phase. So let us see. So this is the cell cycle. This blue one is called as the interphase and black one is the M phase. Interphase is a very long period. Interphase may occupy around 95% of the total cell cycle. Okay, duration of cell cycle. While M phase is a shorter duration, 5 to 7%. Okay, fine. Interphase, for the convenience of study, it has been further divided into three subphases: G1, S, and G2 phase. Duration of cell cycle. Human cell 24 hours. Yeast cell 90 minutes. So duration of cell cycle varies from cell to cell. Fine. Now, interphase. cell enlarges its size duplicates its contents and gets ready to divide. Okay, this is called as the interphase. Three sub phases, G1, S and G2. So let us see. See first, G1 phase. G1 phase corresponds to the interval between mitosis and initiation of DNA replication. Fine. Why it is said that? So see here. This is the end of M phase, mitotic phase. And during this S phase, there is DNA synthesis. And between the M phase and the DNA synthesis phase is the GN phase. That is why it is called as gap 1 phase, means gap between mitotic phase and DNA synthesis phase. That is why gap 1 phase. Fine? It is fine, gap 1 phase. During GN phase, the cell is metabolically active and continuously grows. Okay? Because the cell has to reach the size of the mother cell. It grows on continuously, but there is no DNA synthesis, does not replicate DNA. Fine? Okay. And also during S phase, during G1 phase, most of the cell organelles duplicate. So G1 phase, increase in the size of the cell, most of the cell organelles duplicate. Okay? Most of the cell organelles duplicate. Fine. Second is the S phase or synthesis phase. It is called a synthesis phase because during this phase there is DNA replication. So if the amount of DNA at G1 was 2C, by the end of S phase it will become 4C phase. Marks the period during which DNA synthesis occurs or replication takes place. During this time, amount of DNA per cell doubles. If the initial amount of DNA is depend 2C, then it becomes 4C. But remember, chromosome number does not increase. However, there is no increase in the chromosome number. So if the cell had deployed a 2n number of chromosomes, at G1, even after S phase, the number of chromosomes remains same. So amount of DNA doubles, but chromosome number does not change. Chromosome number remains same. So this is all. And during this phase, there is also synthesis of histone protein. Okay, because in eukaryotes, you know, DNA combines with the protein, histone protein to form chromosome. So there is also synthesis of histone protein. Fine. Next. And in animal cell, during this phase, there is duplication of the centriole. In animal cell during S phase, DNA replication begins in the nucleus and the centriole duplication in the cytoplasm. So DNA replication in the nucleus will be in both plant and animal cell. In animal cell, additional thing is that in the cytoplasm there is duplication of the centriole, which will be used during the spindle apparatus formation. Fine. Okay. Now coming to G2 phase. During G2 phase, there is synthesis and preparation for mitosis. Cell growth continues. So there will be RNA and protein synthesis during G2, but no DNA synthesis. So DNA synthesis takes place only in the S phase. Okay. Next. Now one more thing. If you see this diagram carefully, there are certain cells which very rarely divide. So if the cell decides I have not to divide further, by the end of G1 phase, it will exit the cell cycle, come out of the cell cycle. And this phase is called as G0 phase or quiescent phase. So there are certain cells which very rarely divide. If the cell has not to divide further, by the end of G1 phase, it exits the cell cycle and enters a suspended phase called as G0 phase. Why suspended? I will tell you later. Now the cell in G0 phase is not dividing, but it does not mean it is not metabolically active. So cells in G0 phase do not divide, but they are metabolically active. They will perform metabolic function. For example, heart cells, they are in the permanent G0 phase. Now why we are calling it a suspended phase? Suspended because the cell which has entered G0 phase may never re-enter the cell cycle. For example, heart cell. Or if requirement, it may re-enter. So it is a suspended phase. It can re-enter, it cannot re-enter. For example, if you see de-differentiation, permanent tissue, primary permanent tissue when convert into secondary meristem, it means during the differentiation process, they exited the cell cycle, but during de-differentiation, they re-entered the cell cycle. So this is very good example that it may re-enter or it may not re-enter. See here. So question from this are very frequently asked. Some cells in the adult animal do not appear to exhibit division, and many other cell divides only occasionally as needed to replace cell have been injured. These cells that do not divide further exit G1 phase to enter an inactive state called as quiescent stage or G0. Cell in this stage remain metabolically active but no longer proliferate, means divide, unless called or do so depending on the requirement of the organism. See, heart cell never divide, but primary permanent tissue may start dividing and become secondary meristem. In animals, animals generally mitosis takes place only in diploid cell. But in plant, there are two phases of life. A haploid gametophytic phase, that is also multicellular. A diploid sporophytic phase, that is also multicellular. So mitosis takes place for the haploid cell also for the growth of gametophyte, and in diploid cell also for the growth of sporophyte. There are some exceptions in animal also. In animals, if you remember, the males of honeybee are formed by parthenogenesis. So males are haploid. So growth of honeybee males takes place by mitosis in the haploid cell, which is an exceptional case. Okay, fine. See, however, there are few exceptions in where haploid cells divide by mitosis. For example, main honeybees. Fine. Okay. Now coming to M phase. M phase or mitotic phase, it is a continuous process, but for the convenience of the study, we have divided it into 4 subphases: prophase, metaphase, anaphase, and telophase. During prophase, prophase is marked by initiation of condensation of chromosomal material. Remember, during interphase, if you see the nucleus during interphase, it is in the form of a tangled mass of chromatin, which is called as chromatin reticulum. Why? Tangled mass of chromatin. Now, what happens during the start of the M phase, that is during prophase, this chromatin untangles and it condenses. And when it condenses, you are able to see individual chromosomes like this. Fine. Okay. So the condensation of the chromatin material, the chromosomal material becomes untangled during the process of chromatin condensation. Tangling means chikkulu, untangle, unchikkulu. Okay. The centrosome, which had undergone duplication during S phase of interphase, now begins to move toward the opposite poles of the cell. The completion of prophase can be marked. See, when we will say prophase is completed, chromosomal material condenses to form compact mitotic chromosomes. Chromosomes are seen to be composed of 2 chromatids attached to each other by centromere. This is called as the sister chromatids. So the 2 sister chromatids are attached to each other by centromere, and on both sides of centromere there are proteinaceous plates that are called as kinetochore. Fine? Okay. Second, centrosome, which had undergone duplication during interphase, begin to move towards opposite poles of the cell. Each centrosome radiates out microtubules called as aster. The 2 aster together within the spindle fiber form the mitotic apparatus. See here, what is the meaning of this? Suppose this is a centriole, and microtubules radiate out from the centriole. So this looks like sun. For sun, we use the term aster. That is why this structure is called aster. Now, this aster consists of 2 parts. The microtubules that radiate out are called as astral rays. Astral rays. And this is the centriole. So centriole plus astral rays are together called as aster because it looks like sun. And these astral rays will form the spindle apparatus during the metaphase period, during the metaphase period. Fine. So this is prophase. And by the end of prophase, nuclear envelope, nucleolus, and mitochondria, these endoplasmic reticulum, they disappear. See here. Cells at the end of prophase, when viewed under the microscope, do not show Golgi apparatus, endoplasmic reticulum, nucleolus, and nuclear envelope. These are all gone. Fine. So you see early prophase, and this is late prophase. You see nuclear envelope is disintegrating and chromosomes have become more condensed. And this is transition from prophase to metaphase. So spindle apparatus formation has started. Second is metaphase. In metaphase, so when the nuclear envelope is completely disintegrated and the chromosome condensation is completed, then we say it is the start of the metaphase. So see here, the complete disintegration of the nuclear envelope marks the start of the second phase of mitosis. Hence, the chromosomes are spread throughout the cytoplasm of the cell. By this stage, condensation of chromosome is completed and they can be observed clearly under the microscope. So more the condensed the chromosome, more will be its thickness. That is why, and more better will be the visibility. That is why to study the morphology, the number, size of the chromosome, best stage is the metaphase. Okay, so in metaphase stage, different features of the chromosome are studied because they show maximum condensation. Fine. Second thing is the spindle. So you see that conclusion of the metaphase. First, spindle fibers attached to the kinetochore of the chromosome. Chromosomes are moved to spindle equator and get aligned along the metaphase plate through spindle fiber. See, so first of all, spindle apparatus is completely formed and all the chromosomes, they get aligned at the equator. This is called as metaphasic plate. So metaphase, completion of the condensation of chromosome, completion of the formation of a spindle apparatus, and all the chromosomes aligned at the equator of the cell, which is called as metaphasic plate. This is the end of the metaphase. Now comes the anaphase. Anaphase, during anaphase, the sister chromatids of the chromosome, they split from the centromere. And now they separate. Until they were together, they were the sister chromatids of one chromosome. And when they get separated, they form the two daughter chromosomes. So they will form two daughter chromosomes. So the cell which had 4 chromosomes, now it will have 8 chromosomes. Okay, because each chromosome splits into 2 daughter chromosomes. So see here. See. 1, 2, 3, 4, 5, 6, 7, 8. Okay. So anaphase, centromeres split and chromatids separate, and second chromatids move to opposite poles, and these chromatids will move to opposite poles. Now when the chromatids are moving to opposite poles, see here. So this is the centromere, and these are the arms. So arms form the leading end, they are leading forward. Sorry, centromere forms the leading end. Centromere is leading and arms are trailing back. This is somewhere written here. I'll show you. See, as each chromosome moves away from the equatorial plate, the centromere of each chromosome remains directly toward the pole and hence are the leading edge, while the arms of the chromosomes trailing behind, okay, they form the trailing edge. Thus anaphase stage is characterized by, so what are the features of anaphase? Centromeres split and chromatids separate. Okay, the 2 chromatids separate. And second, chromatids move to opposite poles, or rather you can say the daughter chromosomes move to opposite poles. Fine. Okay. Now telophase, children, is the last phase of the M phase karyokinesis, because remember M phase also has 2 things. Division of the nucleus, which is called as karyokinesis, division of the cytoplasm, which is called as cytokinesis. So right now we are studying the karyokinesis of the M phase, that is division of the nucleus. Fine. Now we are coming to the end of the karyokinesis, which is called as telophase. Now telophase is almost opposite to prophase. Remember, during prophase, the nuclear membrane disappeared. During telophase, nuclear membrane appears. During prophase, there was condensation of the chromatin to form chromosomes. During telophase, there is decondensation of the chromosome to further form chromatin. That is why in telophase, individual chromosomes you cannot see. Fine. So let us see the features of telophase. Chromosome cluster at the opposite spindle poles and their identity is lost as discrete element. Discrete means independent element. Why? Because they have undergone decondensation and again tangling. Nuclear envelope develops around the chromosome clusters at each pole, forming two nuclei. Now one cell has two nuclei. And nucleolus, Golgi complex and E are reformed, which disappeared in the prophase. That is why I said the telophase is almost opposite of the prophase. So with this we complete the karyokinesis. But from one cell to daughter cell we formed when there will be cytokinesis also, cytoplasm will also divide. So this is called a cytokinesis. Now cytokinesis in plant cell and animal cell is slightly different. What you are seeing here is the cytokinesis in the animal cell. What happens? From the center, the cell membrane invaginates. Okay, it forms a furrow. This is called as furrow. Okay. From the center, the cell membrane invaginates, forms a furrow. And this furrow keeps on becoming deeper and deeper. And ultimately a time comes when it divides into two daughter cells. So this is called as cleavage furrow formation method. This is the method of cytokinesis in the animal cell. But remember, in plants, there is a rigid cell wall outside that will not form any furrow. So that is why in a plant cell, there is a different method of cytokinesis. In plant cell, what happens? When the two nuclei form, a cell plate is formed here in the center, a floating cell plate. This cell plate is formed with the help of the vesicles released by the Golgi apparatus. So Golgi apparatus release vesicles. These vesicles used to form a cell plate in the center which floats in the center. Now there is deposition of calcium and pectin on the cell plate. As a result, it keeps on growing towards the outer side and ultimately it forms the middle lamella between the two cells. As a result, the cell divides. So in plant cell, cytokinesis is by cell plate method, while in animal cell, cytokinesis is by cleavage furrow method. This is the difference in the cytokinesis of plant cell and animal cell. Now, last we are coming to significance of mitosis. First, mitosis usually results in production of diploid daughter cells with identical genetic comment. Due to this, the genetic constitution of all the cells of the body are similar. Second, the growth of multicellular organism is due to mitosis. Second point. Third, cell growth results in disturbing the ratio between the nucleus and the cytoplasm. In any cell, there is a fixed ratio between the nucleus and the cytoplasm. So when cell enlarges in size, this ratio between the nucleus and the cytoplasm is disturbed. To restore this ratio, the cells divide, which is called as nucleocytoplasmic ratio or karyocytoplasmic ratio. To restore this ratio, the cells divide. Okay, fine. It is therefore becomes essential for the cell to divide to restore the nucleocytoplasmic ratio. Okay. Fourth point. A very significant contribution of mitosis is cell repair. Suppose there is damage to cells somewhere, so due to mitosis, those dead cells are replaced by new cells. The cells of upper layer of the epidermis, cells of lining of the gut, and blood cells are being constantly replaced. You know that there is a fixed lifespan of our RBC. Okay, there is a fixed lifespan of RBC, it has to be replaced. The skin cells are constantly being replaced. Gut lining, because you know acids are released during or enzymes are released during the digestion, so they cause loss of the gut lining cells. All these have to be constantly replaced, and that can be done only by mitosis. Okay, fine. Next, mitotic is given in the meristematic tissue. The apical and the lateral cambium results in continuous growth of the plant throughout the life. So why plants are able to grow throughout the life? Because there are meristematic cells at the apex and then the lateral position. Due to apical meristem, primary growth takes place throughout the life, and due to lateral meristem, secondary growth or growth takes place throughout the life. So children, with this we have completed mitotic cell division. Now we are coming to meiosis. Just 2 minutes. It is a type of cell division where a diploid mother cell meiocyte divides to form 4 haploid daughter cells. Second, it involves 2 divisions. Meiosis first or reductional division and meiosis second or equational division. But DNA replicates only once. No DNA replication between meiosis first and meiosis second. Fine. So before the meiosis first there is DNA replication, but between meiosis first and meiosis second there is no DNA replication, so it divides only once. Now meiosis first. Meiosis first. Okay, very long duration divided into 5 sub phases. Okay, see, first one is called as leptotene. See here what happens in leptotene. Chromosomes become gradually visible under the light microscope. You are able to see individual chromosomes. Fine. The compact means condensation of the chromosomes continues throughout the leptotene. This is followed by the second stage, that is zygotene. During zygotene, what happens? Pairing between homologous chromosomes takes place, and this pairing takes place due to formation of a structure called as synaptonemal complex. With the help of this structure, synaptonemal complex, pairing between homologous chromosomes takes place, and this phenomena is called as synapsis. Okay, this phenomena is called as synapsis. And the paired homologous chromosomes are called as bivalents. Okay, bivalents or tetrads, because 2 chromosomes, each chromosome has 2 chromatids, so it is called as tetrad. So see here, during this stage, chromosomes start pairing together, and this process of association is called synapsis. Such paired chromosomes are called homologous chromosomes. Each electron micrograph of this stage indicates that the chromosome synapsis is accompanied by the formation of a complex structure called as synaptonemal complex. The complex formed by a pair of synapsed homologous chromosomes is called as bivalent or tetrad. However, tetrad more clearly becomes visible in pachytene. So third is pachytene. Okay. So third is pachytene. During pachytene, first of all, the tetrad condition becomes clearly visible. Second, during pachytene, a very important phenomena takes place, which is called as crossing over. Crossing over means exchange of chromatid segments between non-sister chromatids of homologous chromosomes. Suppose this is one chromosome. These 2 are sister chromatids. This is another chromosome. These 2 are sister chromatids. These 2 are homologous chromosomes. 2 blue are sister chromatids of one chromosome, 2 pink are sister chromatids of one chromosome, and these 2 are the homologous chromosome. But 1 pink and 1 blue are the non-sister chromatids of homologous chromosomes. Between them, there is exchange of chromatid segment, which is called as crossing over. Due to this blue will come here and this pink will come here. Okay. So due to this, new combination of genes is formed, which is called as genetic recombination. So due to this crossing over, there is genetic recombination or formation of new combination of the genes, which ultimately leads to variation. So see, during this stage, 4 chromatids of each bivalent chromosome become distinct and clearly apparent as tetrad. First thing. Second, this stage is characterized by appearance of recombination nodules. Now this crossing over takes place due to formation of an enzyme complex called as recombination nodule. Okay, the site at which crossing over occurs between non-sister chromatids of the homologous chromosomes. Crossing over is the exchange of genetic material between 2 homologous chromosomes, as you can see it here. Fine. Crossing over is also an enzyme mediated process. So where is the enzyme present? Enzyme is present in this recombination nodule. And the enzyme involved is called as recombinase because it is causing genetic recombination. Crossing over leads to the recombination of genetic material of the 2 chromosomes. Recombination between homologous chromosomes is completed by the end of packaging, leaving the chromosomes linked to site of the crossing over. Fine. So, okay, at the site of crossing over. Next is diplotin. So till crossing packaging, the non-sister chromatids of homologous chromosomes were attached to each other. Once packaging is completed, crossing over is completed, the synaptonemal complex dissolves. So due to dissolution of the synaptonemal complex, the homologous chromosomes, which were very close to each other, they start separating from each other. But at those places where crossing over has taken place, they still remain attached to each other, and it is seen as a cross-like or X-like structure, which is called as chiasmata. So let us see. The beginning of diplotin is recognized by the dissolution of the synaptonemal complex and the tendency of the recombined homologous chromosomes of the bivalent to separate from each other, except at site of crossing over. These X-shaped structure are called as chiasmata. And in oocytes of many vertebrates, this diplotin stage can continue for months or even years. Okay, it will not further exceed, but other places this diplotin is completed and it moves to other stage. So if you will see the chromosome at diplotin stage, it will something look like this. They will something look like this. Okay. This structure is called as chiasmata. So this is the places where crossing over has taken place. That is why this X-like structure is called as chiasmata. So this is diplotin. Fine. Next, diakinesis. The final stage of meiotic phase is known as diakinesis. So during diakinesis, the chiasmata which were in the intercalary position, they shift towards terminal position. This is called as terminalization. So shifting of the intercalary chiasmata into terminal position, this is called as terminalization. This takes place in the diakinesis. This is marked by termination of chiasmata. During this phase, the chromosomes are fully condensed and meiotic spindle is assembled. Okay, spindle is assembled to prepare the homologous chromosomes for separation. By the end of diakinesis, the nucleolus disappear and the nuclear envelope also breaks down. It means we are coming to the end of prophase 1. So that is why nuclear envelope breaks down. Diakinesis represent transition to metaphase. So prophase 1 completed, metaphase 1 started. See here. Okay. Metaphase 1. The bivalent chromosomes aligned on the equatorial plate, the microtubules of the opposite poles of the spindle attached to the kinetochore of homologous chromosome. Let us see this diagram. Metaphase 1. This is metaphase 1. See, due to crossing over, now this sister chromatids had got some part of this chromosome and this has got some part of this chromosome, right? So they arrange at the metaphasic plate like this. This is the metaphase 1. Now the terminal chiasmata breaks and the daughter chromosomes, the two chromosomes are separated. So here, so breakdown of this terminal chiasmata, as a result, these cells, these are separated and they move to opposite poles. This is anaphase 1. Phase 1. Anaphase 1. Fine. So anaphase 1, remember in anaphase of mitosis there is breakdown of the spindle of the centromere. In anaphase 1 there is breakdown of the terminal chiasmata. As a result, the homologous chromosomes separate and move to opposite poles. Fine. Now telophase 1. See, this is anaphase 1 which I have just drawn. Telophase 1, you can see here. The nuclear envelope reappears. As a result, two nuclei are formed within the telophase 1. The nuclear membrane and nucleus reappears, cytokinesis followed by, this is called as dyad of cells. After this telophase, where there is cytokinesis. So this is the cell after cytokinesis. So from one mother cell, two daughter cells have been formed and the number of chromosome each daughter cell is half as compared to the mother cell. So remember, mother cell had 4, each cell has daughter cells as 2, that is why we call it as reductional division. And two daughter cells are formed which is called as dyad. Fine? Okay, fine. So this is telophase 1. So this marks the meiosis first. Then between meiosis first and meiosis second, there is a duration which is called as the interkinesis. During interkinesis, there is synthesis of RNA and protein. So during interkinesis, there is synthesis of RNA and protein, but there is no DNA synthesis in interkinesis. Okay. This, and after that comes the meiosis second or equational division. So if you see meiosis second or equational division, it is just like mitosis. See, it is just like mitosis. The two daughter cells, the two cells, chromosomes will arrange on the equatorial plate. They will split from the centromere and each daughter cell will have 2 chromosomes. So you see, mother cell had 2 chromosomes, daughter cell has also 2 chromosomes. So that is why meiosis second is called as equational division. So by the end of meiosis, from one mother cell, we get 4 haploid daughter cells. Mother cell was diploid having 4 chromosomes, each daughter cell is haploid having only 2 chromosomes. Fine. So that is why this reduces the chromosome number. So what is the significance of meiosis? Let us try to see. Meiosis is the mechanism by which conversion of a specific chromosome number of each species is achieved across generation. Okay, they say due to meiosis, in a species, generation after generation, the chromosome number remains same. But you will say, but meiosis is reducing the chromosome number. Then how due to meiosis, the chromosome number generation after generation remains same? See children, if meiosis does not happen, what will happen? Suppose this is a human male. This is a human male. 2n is equal to 46. So if meiosis does not take place, then sperm will also be 2n is equal to 46. And female 2n is equal to 46. If meiosis does not place, then egg will be 2n is equal to 46. Okay. And when there will be fertilization, then the zygote will be 4n is equal to 92. Okay. It means the child will have 92 chromosomes, and the child of this generation will have double the chromosome, 184, and next generation again the chromosome number will become double. But due to meiosis, since sperm is formed by meiosis, so the sperm is n is equal to 23 and egg is formed by meiosis. So this is n is equal to 23. And when the zygote is formed, it is 2n is equal to 46. It means this child will have 2n is equal to 46. So due to meiosis, although meiosis reduces the chromosome number to half, but due to meiosis, a constant chromosome number is restored in a species generation after generation. So this is the first significance of meiosis. Meiosis is the mechanism by which conversion of a specific chromosome number of each species is achieved across generation in sexually reproducing organisms. Even though the process per se paradoxically means just opposite, results in reduction of chromosome number by half. Okay. Second, you see during Paquetine there was genetic recombination. Due to genetic recombination, new combination of genes were produced. Due to formation of new combination of genes, there was variation in the character, and these variations ultimately lead to formation of new species, which is called as evolution. So the basic raw material for evolution is the genetic recombination which takes place during the process. You see here, it also increases the genetic variability in the population of organism from one generation to next. Variations are very important for the process of evolution. So first, it helps in remaining constant chromosome number in a species generation after generation, and second is causes genetic variation which ultimately leads to evolution. So this is all regarding meiosis, and that is all we have right now time for. So wish you all the best. God bless you. Thank you. Bye bye.